welcome to the live solution discussion, paper discussion of NEET 2025 by RV Learning Hub. Today we are going to discuss the whole paper of NEET starting from question number 1 till question number 180. I hope all your paper has been done well. You have prepared for the test for last 2 years, maybe 3 years. All your hard work has come into a fruitful conclusion, hopefully at the end of this NEET 2025. That is my prayer, that is my hope. Today you will get to know about how you have performed after reading and going through the solutions that we have made for you. Before starting the live session of discussion, I would just like to introduce to you the new NEET Replay program that we are going to start. The NEET Replay is a new program launched by RV Learning Hub, which is for the NEET repeaters, for the NEET aspirants of 2026. The aspirants who are planning their NEET 2026, for them we have launched a new program called NEET Repeaters or NEET Replay. Let me brief you about this NEET Replay program very quickly, and after that we'll start our overall paper discussions. Once again, before I start, I just want to tell something about myself. I am Rajai Mithil, Head of the Department of Physics in RV Learning Hub, and I'll be also playing a small part in the discussion of physics paper today. Coming to the NEET Replay program, we have top-notch faculties teaching the NEET content required for the NEET aspirants to clear and become doctors of future that we are hoping for the country. In this program, we will have various study materials which are prepared by our experts across the streams of physics, chemistry and biology, which will be really helpful for the students. The chapters that the teachers will be teaching will be across 97 chapters across the 3 subjects, that is physics, chemistry and biology. Apart from that, we have regular assessments done for the students year long. Throughout the year, we'll have tests being taken for the students, and apart from that, there is a personal guidance which will be given by the experts, the subject experts of physics, chemistry and biology across the year, as well as we will also be talking to the parents throughout the year. The detailed analysis of the PYQs will also be done during the whole course. This NEET long term or the NEET Replay program is going to be done in offline mode in Bengaluru by RV Learning Hub starting from June 2025. The classes will start from June 2025 and the location will be in Bangalore, Yelahanka. So I request all of you, for any queries, just give a call to this number, or there is a link of the website attached in the description of the same video which you are watching right now. Please go to the link, please visit our website, please get in touch with us for any queries. And if your dream is to become a doctor next year and in the next 5 years when you become a doctor, we want you to be one of our students. Hopefully this will turn out to be really good for all of you. Now, not wasting any much of your time, I request Rashmi ma'am and Suresh sir, who are our experts in biology and chemistry, to come with me for the overall analysis of the NEET 2025 paper. I welcome both of them here. This is Rashmi ma'am and this is Suresh sir. Hello, so both of us, all the 3 of us will be discussing the paper, the overall analysis of the paper along with our teams. Coming to the paper, I would just request Rashmi ma'am to start with the analysis of biology, how she feels biology exam was. Ma'am.
Hello students, hope you all have done your NEET exams very well, and we all look forward to our analysis. So not taking much of your time, from a student's point of view, looking at the question paper of biology, children, I think it falls in the moderate to the hard section. Am I correct to most of you? Yes. So it's not an easy paper, it's a little tough. And what is our mantra, children? Yes, it is the NCERT. So looking at all of our questions in the biology section, Those who have gone through line by line of the NCERT definitely will feel it as an easy paper. But for those who have skipped out even a single line, it's going to be a tough one, right? So the overall analysis is, with respect to biology, it is moderate to tough. The second special feature that we are noticing this year is we do not have botany separate section and the zoology section separately. It's been clubbed with botany and zoology questions. So those of you who are expecting first I will solve the botany, then go to zoology, they would be in a dilemma because 91 starts with one section, 92 goes to the next section. So it's a mix of botany and zoology. The third thing, it's a mix of both first year and second year. They have not lowered down the first year syllabus or given more weightage to the second year. So it's a mix and balanced question of both the first year and second year syllabus. All right. So now I will hand over to our Suresh sir.
Before we start the discussion, before we start the discussion over chemistry also, sir, 1 minute, can you give the mic to madam? Uh, do you think there's a reason why the NEET people, NTA people have decided to mix zoology and biology? What my observation is that they want to make this year's NEET paper a little bit of challenge for all the students, which is unlike what happened last time. So is that my observation and is that correct? Do you think so?
Possibility, sir. But the other observation that we should notice, some of the students, they have easier version towards botany, easier version to zoology. So for those kind of students, the moment they see a tough question in botany, it should not loosen down their enthusiasm that I am not able to focus. So maybe that's why they have mixed botany and zoology, so that the students are enthusiastic throughout and they are not held up with botany or zoology. This could be one of the reason.
No, botany, zoology, both are tough or only one particular one?
No, both are moderate to tough.
Hold it, not an issue, hold it.
Hi, good evening to all of you. I hope you have done very well in the examination. So when it comes to chemistry, let me classify chemistry into three parts. As we know, some of the students, they don't like organic chemistry. Okay. Some of the students, they don't like physical. Now, inorganic part, when it comes to inorganic part, some of the students don't like. But overall, when it comes to overall paper, it is going to be moderate to difficult. This is not easy paper. This time, this is not easy paper. Now, some student, they hate inorganic chemistry because that is memory based. Now, this time, I don't know. We used to see the inorganic chemistry. Generally, they ask very simple questions, but this time they have asked very, very hard type of questions. And also, when it comes to organic, this is not easy. It is not easy. So, out of let us assume 15 questions, they have asked 8 questions are moderate. Remaining questions are given as a hard. When it comes to physical chemistry, there are few questions are very easy, but overall physical chemistry also it is given as a very hard. So overall chemistry paper look like very, very hard. It is almost I can say that equal to mains level. So even though it is NCRT based, this time they've asked bit application type. So whoever has done application type, they will able to solve. So I think out of 180, it's going to be very challenging for the children this time. Okay, so that's all about chemistry. So I think, like ma'am said, the botany and zoology it is given as a very hard, are moderate to difficult. Same kind of observation we have seen in the even chemistry as well. Okay, it's chemistry, physics.
Coming to physics, I would like to say that I share the same opinion. Even for physics, the paper is on the difficult side. I would say the paper is on the hard side, not even just moderate, given that the time limit to solve questions is very low. You are expected to solve 45 questions in 60 minutes, and the level of questions and the applications of the questions require you to give more than 2 minutes per question definitely. So yes, the physics paper has been difficult. Looking at the overall paper, I believe that cracking NEET is going to be challenging this time. overall 720 on 720 across India will also be a rare thing. It's not going to be easy as it has been in the last 2 or 3 years. And sharing of the ranks at the top marks, that is 716 or 715 or 710, is also going to be a little rare. Only few students across India will be getting top ranks in top all India rank 100. So 720 marks, if I say 720 marks is rare. That is, if I can sum up the whole analysis this time, 720 is going to be rare. And overall, the NEET paper has been challenging. Now, why am I stating this out and why are we stating this out right now at the start of the analysis is because if you found your NEET paper a little difficult, a little challenging or more challenging, stay relaxed. It was an observation across India for everyone. The paper was in fact very challenging for everyone. All right.
If
So
someone find easy, if someone find it is very easy, so obviously you are the, I think, top notch student. Okay. So I feel, chemistry wise, especially when it comes to chemistry wise, so generally students have a lot of questions like, uh, anyway, like not like physics and botany and zoology. Chemistry wise, we have three parts. Students always worried about sometime organic, sometime physical, sometime inorganic. This time, inorganic chemistry also, we used to expect at least, they'll not go beyond, application type for inorganic chemistry. But even we have to think, this is a kind of memory based question along with application type together they have asked in the examination. We didn't expect every, last year paper when we have seen it looked like, very easy. So like that we have expected this year also. So we didn't expect actually like the type of questions we used to see in the mains level. But this time they've asked even when it comes to stereochemistry in chemistry application type they've asked. Okay, so I think overall paper is I feel very very difficulty. So if someone is finding easy that paper is, I think you'll get very good rank. I hope you all are agree with that.
yes, sir. Yes. Definitely, we agree with that. So, looking forward, if you are looking forward for NEET 2026 or anyone of your siblings or friends are looking for NEET 2026, please tell them that it is going to be tough also if it follows the same trend, and the preparation of NEET should be done at a little more level than NEET level, which has been observed in the last 5 years. So, with that, I would like to conclude the overall analysis of the paper, and I would like to request Rashmi ma'am to take over the first 15 questions of biology to be discussed and analyzed in front of you. So, with that, I think Suresh sir, do you want to add anything?
Yes, sir. I think that's all. I think I will continue with the question paper discussion. I hope this session is going to be very, very helpful for you. Now we just started in 1 hour itself. Okay. You can also check your answer keys. Okay. And also, just see how many marks you are going to get. Okay. If you are getting 600 above, I think you'll surely will get a very good rank. Okay. That's all from my end. Thank you. And so now, ma'am will take over. She will explaining the biology questions. Thank you.
All right, students, now you have got the overview analysis, and also for those of you who are looking forward for the repeaters course, we do have a plan here at our week. Please do come and join us. So without wasting much of your time, children, let's go into the discussion part of biology section, correct? And this is me a little bit, you can see here. All right. Okay, so we are going to discuss the version 48, children. 48 is our version that we will be discussing today. Most of you might have different versions. So I know it's going to be a little difficult for you to look out for the questions. Please do have your question paper with you. For those of you who have got the version 48, the first question itself is a tricky question. Am I right? You all agree? Yes. It is a tricky question, children. The moment you look at the question itself, you might wonder what is this. So let's look into the question. The question says, in the above represented plasmid and an alien piece of DNA is inserted at eco R1 site. Which of the following strategies will be chosen to select the recombinant colonies? White color colonies will be selected. Blue color colonies grown on ampicillin plates can be selected using ampicillin and tetracycline containing medium plate, or the fourth option says blue color colonies will be selected. So what is our answer here? It says an alien piece of DNA is inserted at the eco R1 site, and our eco R1 is having beta galactosidase. So what is beta galactosidase, children? It is an enzyme that is going to break down lactose, correct? So it is going to break down lactose to glucose and galactose, right? And if beta galactosidase is present in that particular plasmid, we are going to see if beta galactosidase is present, we will have blue color colonies. Since our alien piece of DNA is inserted at this particular place, we have the insert. So beta galactosidase is absent, so the colonies will be white in color, correct? So it's a recombinant, correct? A recombinant plasmid. So our answer is going to be option 1, which says white color colonies will be selected. Moving on to the next question. The protein portion of an enzyme is called. The next question is a very easy question. Apoenzyme, prosthetic group, cofactor, coenzyme. Protein portion of the enzyme. The whole enzyme is called as holoenzyme, correct? So holoenzyme has got two parts, the protein part and the non-protein part, correct? The protein part of the enzyme is called as apoenzyme, and the non-protein part is called as prosthetic group, right? So prosthetic group can be your coenzyme or a cofactor. So since they have asked protein part, it is going to be apoenzyme, option 1 again. All right, next question. Given below are two statements. We have two statements. Statement 1 says the primary source of energy in an ecosystem is solar energy. Is it right? Definitely yes. Solar energy is nothing but the sun. So yes. Statement 2 says the rate of production of organic matter during photosynthesis in an ecosystem is called as net primary productivity, which is wrong. Correct? So what is it called, children? It is called as gross primary productivity. Correct? So GPP. So statement 1 is correct, but statement 2 is incorrect. So we go with option 1. All right. Next question. Again, we have two statements here. Correct? The two statements here are a typical unfertilized angiosperm embryo sac at maturity is 8 nucleate 7 celled. Is it right? Definitely yes. So it is a 7 celled 8 nucleated structure. The egg apparatus has 2 polar nuclei. 2 polar nuclei? Wrong. So what is the structure, children, if you remember? This is our embryo sac, correct? So at the chalazal end, this is your micropyle end. This is your polar nuclei at the central cell. So this is your egg apparatus, correct? So egg apparatus is going to have not 2 polar nuclei, it is having 2 synergids, correct? So we would say that assertion is true, but reason is false. Assertion is true, reason is false. All right. Next question. Neoplastic characteristics of cells refer to. Children, this question is as it is in the NCERT if you take cancer. Okay, if you have seen and studied the NCERT line by line, you got this answer right. Neoplasm. What is neoplasm? A mass of proliferating cell. Yes, it's a characterization of cancer cells, a tumor. Rapid growth of cells? Yes. It does invasion and damage to the surrounding tissue? Yes, it is there in the NCERT. Those confined to original location, no. So, these cells can undergo metastasis. Okay. So, our options A, B, C is correct. So, that would be option 4. A, B, C only and not 3, not 4 because they can undergo metastasis. All right. Which one of the following is a characteristic feature of the gymnosperms? Very simple question. They have given us 4 options. Seeds are absent. Gymnosperms have flowers for reproduction. Seeds enclosed in fruits. Seeds are naked. The very characterization of gymnosperms, one word for gymnosperms is they are naked seeded plants. Correct? They do not have a covering called fruit. So, the seeds are naked. Very, very simple question this is. All right. Match the following. We have list 1 with list 2. This is your first PUC, last chapter, functions of the hormones. If you see what progesterone is a part of, it is here with the corpus luteum. Okay. Relaxin is going to be the ovary during delivery process. Relaxin is going to be secreted. Melanocyte stimulating hormone is pars intermedia, and catecholamines is going to be by the adrenal medulla. So, A is going to be 4, relaxin B is 2, C is 1 and D is 3, which is our option 3. All right. Okay. Which chromosome in the human genome has the highest number of genes? A very simple question again. So, chromosome 1, 10, X and Y, highest number of genes is definitely the chromosome 1 and the least is going to be the chromosome Y. So, this shares the least number of genes. Okay. Which of the following statements about rubisco is true? Our very important photosynthesis chapter. It is an enzyme involved in the photolysis of water. No. So, rubisco is carboxylase oxygenase, right? So, what is it going to do? It is going to have higher affinity for carbon dioxide and not the oxygen, correct? So, this option becomes the wrong option because it has higher affinity for carbon dioxide. It is active only in the dark? Definitely no. So, the right answer is going to be it catalyzes the carboxylation of RUBP, right? Okay. The first menstruation is called, a very simple direct question of your second PUC syllabus. The first menstruation, diapause, no, ovulation, no, menopause, no. Menopause is the last reproductive cycle, correct? So, the first menstruation is called as menarche. Very simple question. All right. Which one of the following genetically engineered organisms was used by Eli Lilly to prepare human insulin? We have virus, phage, bacterium and yeast. A very simple question again, children, from biotechnology applications. You have the diagram based question here. So, Eli Lilly used E. coli, correct? So, what is E. coli, children? It's a bacterium, correct? So E. coli is a bacterium and the right answer is option 3. Okay, next. Given below are two statements, assertion and the reason. Let us see the assertion here. It's from the animal kingdom. Assertion says all vertebrates are chordates, but all chordates are not vertebrates. Right? Yes. Reason says the members of subphylum Vertebrata possess notochord during the embryonic period. The notochord is replaced by a cartilaginous or bony vertebral column in the adults. Is it right? Definitely yes. Correct? So both are statements, assertion and reason is correct. And if you look at the correlation, children, here they say that notochord is replaced by the vertebral column in adults, which is why all vertebrates are chordates, but all chordates are not vertebrates. Correct? So we would say that both A and R are true, and the reason is the correct explanation for the assertion. All right. Next question. What is the main function of the spindle fibers during mitosis? To repair damaged DNA, to regulate cell growth, to separate chromosomes, to synthesize new DNA. Those of you who do not know what is spindle fibers, this is the cell, right? And we have the spindle fibers formed here during the cell division, correct? So what these spindle fibers are going to do, children? They are going to pull the chromosomes apart during the anaphase, correct? Yes. So the right answer is option 3, to separate the chromosomes. All right. Match list 1 and list 2. We have the scientists at one end and their discovery is a very simple question again. Alfred Hershey and Martha Chase. The moment you hear Hershey and Chase, what did they do? The first experimental proof that DNA is the genetic material. Euchromatin. What is euchromatin? It is a loosely packed, light-stained chromatin. Frederick Griffith worked with Streptococcus pneumoniae. Heterochromatin is a densely packed, dark-stained chromatin. So A is 4, B is 3, C is 1 and D is 2, which is option 1. All right. Okay. I think we are at the last part, last question of my analysis for biology. The list 1 with list 2, we have adenosine here. What is adenosine? Adenosine is a nucleoside, nitrogenous base plus a sugar. Adenilic acid is a nucleotide. Nucleoside plus a phosphate group is a nucleotide. Adenine by itself is a nitrogenous base, correct? And alanine is an amino acid, very simple. So A3, B2, C1 and D4. Thank you students for listening. Hope you got the answers for the first 15 questions of biology, right? So now I invite Suresh sir to take up the next session and solve the first part of chemistry. Thank you.
Thank you, ma'am. So it was a wonderful presentation from biology team. And I would like to introduce myself, Dr. B. Suresh Babu, and presently working as head of the Department of Chemistry at RV Learning Hub. And first 15 questions from chemistry I'll be discussing. The remaining questions, my team, the remaining two faculties will discuss that. So let's discuss about first 50 questions. Okay, the first question says that here, identify the suitable reagent for the following conversion. So the reactant is given here is ester. Ester is converted into aldehyde. If I look at the option, sodium borohydride generally will not reduce ester, so we can avoid this first option. Now the second one, hydrogen palladium barium sulfate is given. This is generally used for hydrogenation reaction Anyway, hydrogenation also cannot be done with the, for the ester. The next third one, lithium aluminum hydride is very strong reducing reagent. It will reduce easily the ester into alcohol, not aldehyde. Now finally we have, ALH iso butyl twice is given. Students may have a confusion here. This is actually dibol H. Dibol H is a reagent which convert the ester molecule into directly benzaldehyde. We have one option now, the fourth one. That's a very simple question, but student may get confusion. They have given different formula, ALH IBU twice, diisobutyl aluminum hydride. This will generally transfer H minus and now this H minus will attack and here carbonyl group, and then OCH3 get eliminated, directly will get benzaldehyde. Okay. Next, go to the next question. The correct order of decreasing acidity of following aliphatic acid. Whenever it comes to acidity, acidity depends upon the conjugate base. If conjugate base is stabilized by electronic factors, now the conjugate base is required electron withdrawing group, not electron donating group. Now if I look at these options, the first option, if you see the HCOOH, now CH3COO minus. Let's take here conjugate base or C double bond O minus. Now conjugate base must be stabilized by electron withdrawing group. But if you look at this option, all are given as the electron donating group. That means more electron donating group, it will destabilize conjugate base. Now first case, if I take now H bond, it's not item. Okay, so HCOOH generally, HCO minus generally generated. Now CH3COOH generally, which gives you CH3COO minus, and similarly the next molecule also which gives you CO minus. If I look at this option, I can take first option itself. The reason is this will have only hydrogen, this molecule has only CH3, but if you see from hydrogen to CH3 there are two CH3, again this is converted into trimethyl group. So here electron donating groups are keep on increasing from formic acid to this acid. So electron donating group generally will decrease its conjugate base strength. So here instead of going for all option, I can take directly first option here. Okay. Okay, I'll go to the next question. Okay. I think it got stuck. Only the molecules are highly or strong acidic in nature. Now whatever is given here, so first one here, the formic acid, and this conjugate base is now HC double bond O minus. Now if I take one more CH3 C double bond O minus here, the plus I group is added to the conjugate base. So whenever plus I groups are added to the conjugate base, that is destabilizing. So if I look at this option, formic acid, there is only one hydrogen, and if I take acetic acid, there is only one methyl group, and if I take other molecule, that you can see there are two methyl groups is added to the COOH. So conjugate base stability decreases when we add more electron donating groups. So obviously I can take first option instead of going for other options, I can take directly first option. Now we'll move on to the other question. Which one of the following reactions does not belong to Lassine test? Lassine test generally which is related to, okay, the nitrogen and also sulfur and also halogen reduction generally we will do. So detection of these elements, which comes under qualitative analysis of organic compound. Now if you look at this one, sodium and halogen, this is used for Lassine test. Now cupric oxide with a carbon, this is actually, identification of carbon and hydrogen. Okay, so that is why we can take directly as a, the estimation, identification of carbon and hydrogen is given. But when it comes to third one, this is also used for, identification of nitrogen, and this is used for sulfur. So finally, the only one option we have, that is second one. Okay, next question. If the rate constant of a reaction is 0.03 second inverse, how much time does it take for 7.2 mole per liter initial concentration is given? Now, the concentration of the reactant to get reduced into 0.94 mole per liter. Okay, so but you may have a lot of doubts here. How do we know that is what kind of order? It's easy by looking at this question. There are units are given. If you look at this one, second inverse generally used for first order. So directly we can take first order equation. So K, that is equivalent to 2.303 by T log R naught by R, where R not equivalent to initial concentration, where R equal to change in concentration. Now T is time. How much time? Anyway, in the question they have asked how much time does it take, and the rate constant already they have mentioned 0.03. Now if I substitute everything, 0.03 that is equivalent to 2.303 T we have to find out, and log or R naught value is given here 7.2 mole per liter, and it is reduced into 0.9 mole per liter. Directly I can take here 0.9. So if I simplify this entire calculation, and you will get final answer 69.3 seconds. So everything is given here, 7.2 by 0.9, that will give 8. So the log 8 can be written as 2 cube. So 3 log 2, and log 2 value we know 0.30. And if we substitute everything in this equation, if we simplify this, and the final answer is 69.3 seconds is correct answer. Okay, this is related to physical chemistry, that is chemical kinetics. Okay, so we'll move on to the next question. So given below are two statements. Statement 1, a hypothetical diatomic molecule with bond order 0 is quite stable. So if bond order is equivalent to 0, that is always unstable because there is no bond between two atoms. So obviously this is going to be incorrect statement. Okay, incorrect statement. Next, as the bond order increases, bond length increases. That is going to be wrong. So when bond order increases, bond length generally decreases. This is again false statement or incorrect statement. So if I look at this option now, statement 1 and statement 2 both are false. For this question, answer is 4. Okay. Next question. Out of the following complex compounds, which of the following compound will be having minimum conductance in the solution? There is ambiguity also. There is bit ambiguity. So if I look at this first one, the molar conductance or the conductant depends upon the ions in the solution. Okay. Now if you look at this one, this is plus charge and here 3Cl minus. There are 4 ions are liberated in this solution. If I take second one here, CONH3 5 times and Cl, the whole bracket, that can be written as 1 cation, the other one is anion. This will produce 2 ions here, but when it comes to 3 and 4, here there is no conductance, 0 conductance I can take, because this molecule will not liberate any kind of ions. So this is going to be 0 conductance, this is also 0 conductance, and if you look at this one, first option, you see maximum ions, that means this will have higher or the maximum conductance, but they are asking minimum conductance. If they ask you 0 conductance, any of these 2 options, like 3 or 4 option we can take, but they are asking minimum conductance. So I will go for second option, not first option. Okay. Next, moving on to the next question. Now which of the following aqueous solution will have highest boiling point. It depends upon the want factor and the concentration. This is related to physical chemistry solution chapter. Solution chapter. Now, if you look at the concentration is given and the total number of ions are produced by Na2SO4, there are 3. One is 2 sodium plus ion, one is SO4 minus 2 ion. So, this is 3, 0.01 into 3, that will give 0.03, that is molar concentration. Now, this is glucose or fructose, I can say, because they have given only formula. So, generally they will not undergo any ionization. So, that means it will exist as only one molecule. So, that means 0.015 into 1, that will give you again 0.015 only, right. Next, if I take now 0.01 molar into urea, that is also will give same kind of concentration, because there is no change, because urea will not undergo further dissociation. Next, if it comes to the fourth option, 0.01 molar concentration is given, and potassium nitrate, there are 2 ions. Now, the concentration 0.01 into 2, that will give you 0.02 here. So, if you see, if you look at the 4 option, the one with the highest concentration, that is always will have highest boiling point. Now, I will go with the first option, that is 0.01 molar sodium sulfate. This is related to solution topic in the second year, and we will move on to the one more question here. Given below are 2 statements. One is labeled as assertion A and other one is labeled as reason. So, what they have given here, both are primary alkyl halide. Okay, this is related to halogenated hydrocarbons. Now, this one is primary alkyl halide. This is also primary alkyl halide. Generally, in SN2 reaction, the methyl halides are more reactive. Then it comes to after that, primary, then followed by secondary, then followed by tertiary alkyl halide will come. Now, if you look at this option, iodine generally act as a better leaving group because of atomic size is very bigger. If I take C bond iodide, iodine atomic size is very bigger. So, iodine easily can leave from the carbon atom. Now, if I look at the C bond Cl, generally chlorine atomic is small atomic size as compared to iodine. So, leaving group ability is bit less. So this assertion is correct. In SN2, iodoalkanes are more reactive than the chloroalkanes. Assertion is correct. Now, iodine is better leaving group because its large atomic size. So, this is also correct. So, both are correct, and I can take here both A and R are true, and R is the correct explanation of A. So, I would prefer 3 option. Okay. So, moving on to the next question. Consider the following compounds as compared to other question. This question is very easy. So, most of the students looking for this type of questions. So, if you look at this one, potassium with a O2 is given. This is basically peroxides, right? You know, right? The first group and second group elements generally form monoxides or dioxides and peroxides also. Now, this will come under peroxide bond. Now, K generally exists here +1, oxygen will have -half. Okay. Now, when it comes to hydrogen peroxide, oxygen has -1, hydrogen anyway it has to be +1. So, when it comes to H2SO4, the 2 hydrogen mean 2 + 1, and 4 into oxygen, 4 into -2 will give -8. To balance this equation, sulfur has to be +6. So, the one is +1, one is -1, other one is +6. So, +1 and -1 and +6 generally is given answer. Okay. The next question, this is also very easy. So, Haber process, we have to match the list 1 to list 2. Haber process is very simple. We require iron catalyst. This is the memory based and directly we can remember and can answer. The Walker process is very important. Okay, Walker process generally the conversion of alkenes into aldehyde. We require palladium catalyst. Palladium chloride undergo generally oxidation. This will not generally kind of catalytic mechanism. Okay, so Walker oxidation require palladium chloride and Wilkinson catalyst. Okay, Wilkinson catalyst is basically this one. This sometime also will be used for hydrogenation purpose. Okay, so the Wilkinson catalyst is triphenyl phosphine, tris triphenyl phosphine rhodium chloride we used to call it. And Ziegler-Natta catalyst generally used for what? Polymerization reactions. So what is required here? Titanium chloride, aluminum, chloride, or you can take, trialkyl aluminum also you can take. So what is given here? The A is 1, okay, B is 2, C is 3, and D is 4. So I prefer here only first option. Okay. Next, we'll move on to the next question. So this is a very memory based question, related to periodic table as discussed, just now. There are some inorganic chemistry memory based questions they've asked. So, like you should have gone thoroughly with the periodic table and their properties, physical properties and chemical properties, then only you can able to answer this. Now, the statement 1 says that here, like a nitrogen that can form ammonia. So basically what they are telling, nitrogen will form ammonia, and there is a arsenic. I'll take arsenic here. Now it will also form arsine. Arsine is basically AsH3. Even this is also can form. So statement 1 is correct statement. There is no issue with that. And antimony cannot form antimony pentoxide. That is wrong. Antimony also can form pentoxide, that is Sb2O5. This is balanced formula. So this is false statement. So statement 1 is true, statement 2 is false. Now if I take now statement 1 is correct, but statement 2 is incorrect. So I will take this one as first option. Okay. Next, moving on to the next question. The ferromagnetism is considered as an extreme form of paramagnetism. That is absolutely correct. That is memory based and is conceptual based. There is no much explanation for that. And when it comes to this statement is correct. When it comes to statement 2, the number of unpaired electrons in chromium plus 2 ion. If I take chromium configuration, chromium is atomic number is 24, scandium, titanium, iridium and chromium, and argon I can take here, and 4s2, 3d4 I will take. Now what they have given here is Cr plus 2, and that means what? Loss of 2 electrons will give you Cr plus 2. So Cr plus 2 means here argon and 4s0 and 3d4. There are 4 unpaired electrons are present in chromium. Now what they have given here, neodymium, okay. So this is basically Nd. How to write this formula? Xenon, and you have to go for with 4f4 and followed by 6s2. This is the neodymium, the general formula or electron configuration. Now again what they have given here, Nd plus 3 means loss of 3 electrons. If I remove 3 electrons here and xenon and 4f, I can take 3 here, and this is going to be 6s0. Here total number of unpaired electrons are 3. Here 4 unpaired electrons. What they have asked here, the number of unpaired electrons in chromium plus 2 is the same that of neodymium plus 3. This is very hard question for you. Actually it's very difficult to answer because you should know the formula. Okay. Now here this statement is incorrect. Now if I take option now, statement 1 is true and statement 2 is false. So here which option I can take? Yeah, first option. Statement 1 is true but statement 2 is false. Okay. Next, moving on to the another question. Which one of the following reactions does not give benzene as the product? Does not give benzene as the product. Okay. Now let's take a first example here. Sodium benzene. Okay. Sodium benzoate is given basically. Soda lime is used for what? Decarboxylation. Okay. Decarboxylation, removal of carbon dioxide. So I'll remove this one and followed by you do hydrolysis, anyway we'll add some kind of hydrolysis. The finally you will get a benzene. Finally you will get benzene. So this is correct. So correct means it will give benzene. Okay. Now N-hexane, molybdenum oxide, or else you can use vanadium pentoxide or chromium oxide. This is used for aromatization. What is the meaning of aromatization? conversion of long chain molecules that will be converted into benzene, that is basically is an aromatic compound. So if I look at this second option, the long chain molecule is given, the N-hexane. N-hexane is converted into benzene, that is correct, because this is undergoing oxidation. So this will give benzene, but what we have to find out, which one will not give benzene. So if I take this one, this is a favorite for all the children here. We keep repeating in the first year and second year also. So which one will produce benzene and which one will produce 1, 3, 5 trimethyl benzene. Okay, this is a very, very simple reaction for the children. And alkyne, the ethyne will be in the presence of red hot iron tube, they undergo cyclic polymerization. Cyclic polymerization, there are 3 molecules we have to take that will give benzene. So 3 option also will give benzene. Finally, 4th option, if you look at this one, benzene diazonium salt halide. So benzene diazonium salt halides are highly unstable in the presence of aqueous medium. So benzene diazonium salt halide in the presence of water, what will happen basically, there is a nucleophile from the water, and here nitrogen is positive charge. This bond will be shifting to the nitrogen side, that will create aryl carbocation, basically benzyl, no, I'm so sorry, not benzyl carbocation, it is basically phenyl carbocation. Now phenyl carbocation is attracted by OH minus like this. So what will happen basically, you will get a phenol, okay, you will get phenol here along with nitrogen gas, Cl minus is eliminated. Now if I look at this option now, so here benzene is formed, okay, here also benzene is formed, here also red hot iron benzene is formed, but here does not give benzene as the product. You may ask one question, sir, benzene is there. No, that is not correct actually. What they are asking, benzene as a product, it is not exactly benzene, it is actually phenol. Phenol. So that's the reason I'll go for 4th option. Okay. I think students are very smart. You may have a lot of questions on this. This is also benzene. They're asking only benzene as a product. Actually, even though it has a benzene part, but cannot be taken as a benzene, so it is basically is a phenol. Okay, phenol is a product that is formed in the 4th reaction. Okay, now let me go through the one more question. So this is related to chemical bonding. So based on hybridization. So every time in NEET examination we see this type of questions are generally common. Okay, I hope we'd have answered this already. If I look at this one, xenon trioxide. So xenon trioxide, xenon is basically belongs to the main 8th group element. So it can form maximum 8 bonds. Anyway, xenon generally form maximum 8 bonds. Now if you look at this one, xenon trioxide, there are already 6 bonds are formed. There will be 1 lone pair. As we know, if you want to calculate for hybridization, we require only sigma bonds or lone pair. There will be 1 lone pair and 3 sigma bonds. There are 3 pi bonds. 3 pi bonds generally we don't consider for hybridization. We have a 3 sigma, 1 lone pair, this will give 4. But anyway, this is sp3 hybridization, and also there is a 1 lone pair. So according to VSC PR theory, when lone pair is present, entire geometry will change. That will give you different shape. So if I look at this xenon trioxide is basically, now I can go for sp3 and pyramidal. sp3 and pyramidal, because 1 lone pair, it is like ammonia, right? You take 1 lone pair, you have 3 sigma bonds, and you will get kind of ammonia structure, that is sp3. For A, I will take here second option, xenon difluoride. Now xenon already forming 2 bonds. Okay, so here 1 lone pair, here 1 lone pair, here 1 lone pair. There are 3 lone pairs and 2 bond pairs. So 3 plus 2 will give 5. 5 must be equivalent to sp3d hybridization. So along with, you can also see there are 3 lone pairs with the 2 bond pairs. This will give you linear structure. So sp3d linear, where is that answer? First option. So I will go for first option here. Now xenon oxyfluorides, basically there are a lot of things we can see. Now if I look at this xenon oxyfluoride, that is tetrafluoride is given. So if I write the main one, so anyway oxygen will form double bond O. The fluorine will form generally here all sigma bonds. So now we can look at this xenon central atom. It has 6 bonds, but there is one more lone pair like this. So one lone pair and 5 bond pairs. That means I can take as a 5 sigma bond, and there will be one pi bond. Anyway, pi bond we don't consider for hybridization. 5 plus 1 will give you 6 here. That must be sp3d2, right? So sp3d2 with a one lone pair, generally that is square pyramidal. So I would prefer here 4th option. Then xenon f6. F6 generally there are 6 sigma bonds, one lone pair. 6 plus 1 that gives 7. 7 that is equivalent to sp3d3. So xenon hexafluoride I would prefer here 3rd option. So if I look at this option now, A is 2, okay, B is 1 and C is 4. So 3rd option is correct. Okay. Now we'll moving on to the next question. How many products including stereoisomers are expected from the monochlorination of the following compound? Okay. Now what is given here is basically we start giving numbering IUPAC name 1, 2, 3, 4. This is basically 2-methyl butane. Okay, 2-methyl butane. So 2-methyl butane is basically undergoing monochlorination. So we have to see first how many equivalent hydrogens are present in the given molecule. So how to identify this one? So these two CH3 are connected to same carbon. These two are equivalent. That means what? You replace any side from the CH3, you will get the same IUPAC name. So now if I look at this second one here B, this is C and this is D. Now you may have a doubt, this CH3 both are equivalent or not. That is wrong. So if you look at this CH3 both are different. The reason is if I replace one hydrogen with a 1 Cl, you will get different IUPAC name. For this also you will get different IUPAC name. Now overall, now let me write here how many monochlorination possible. So I'll start with this here, one compound, and I'll take one more compound like this, and I will take one more compound like this, and the one more compound will be like this. Okay. So and they have mentioned clearly including stereoisomers. So stereoisomers basically required chiral center for the molecule. So if I look at this one molecule, there is a chiral center. Chiral carbon or chiral center is basically carbon should connect with the 4 different groups. So if I look at this one, so this is group is different, this group is different, this group is different, this group is different. That means we know the formula, right? 2 power n, where n equal to number of chiral center, and here 1 chiral center, this will give you 2 isomer. And this one there is no chiral center, this will exhibit as structural isomer. And the next molecule, if you look at this, there is a 1 chiral center for the molecule. So 2 power n, and that will give 2 power 1 equivalent to 2. Again this will give you 2 isomer, and the next molecule will give 1 isomer. So overall if I count along with the structural and stereoisomer, along with that you will end up with here 6 answer, that is second option. Now that is for chemistry, and now I would like to welcome Dr. Garima madam. So she will be doing 15 questions of biology. Okay, so welcome ma'am, and thank you all.
Hello everyone. Thank you, sir. I will be taking the next 15 questions for biology. Let's start with the first one. Which of the following statement refers to as reductionist biology? The word reductionist biology is telling that it is something related to reduction. We are developing or breaking them into smaller fragments to analyze the elements and the composition of the cell. So it is the physiochemical approach. What it is? It is a physiochemical approach to study and understand the living organism. Here, First is the answer that which of the following statement is reductionist biology, breaking them into small elements and then analyzing its composition. So, physiochemical approach to study and understand living organisms. Next question says, after maturation in primary lymphoid organs, the lymphocytes migrate for interaction with antigens to secondary lymphocyte organ tissues like. What is thymus, bone marrow? Thymus and bone marrow are the primary lymphoid organ where they will originate and mature. But for the interaction, they will go to the secondary lymphoid organs which are spleen, lymph nodes and the Pierce patches. So, option C, D, E, they are the secondary lymphoid organs where the antigens will interact. So, let us see which is the option which is correct. C, D, E only, that is option 4 is the correct answer. Next question says, match list 1 with list 2. The evil quadrate. The evil quadrate are the 4 reasons. Quadrate means 4 reasons which are the causes for the biodiversity losses. So, A goes with the third option. Ex situ conservation means conservation of something outside their natural environment. So, cryopreservation goes with this. Lantana camera is an alien species native of America. So, C goes with the 2. Dodo is extinct species. Let us see which is the correct option. A goes with the 3. All the options says A3. B goes with 1. That is fourth option, cryopreservation. C goes with second, alien species, and D goes with extinction. So, fourth option is the correct answer. Let's move on to the next question. How many meiotic and mitotic divisions need to occur for the development of a mature female gametophyte from the megaspore mother cell in an angiosperm plant? Megaspore mother cell will undergo meiosis. It will go one meiosis, and in meiosis one cell give rise to 4 daughter cells. So, here we will get 4 megaspore cells, out of which 3 will degenerate and only 1 will be there. That 1 will undergo mitosis 3 times, 1 time, then it will undergo 3 times mitosis and it will form a 8 cell 7 nucleate stage. So, the correct answer will be 1 meiosis and 3 mitosis. The option which holds correct is the option number 1. The next question says, which of the following type of immunity is present at the time of birth and is a non-specific type of defense in the human body? See, they are telling which type of immunity is already present when a child is born. It is inherited one. The inherited immunity is non-specific, just fighting with the diseases. That is the innate immunity. Cell-mediated immunity is the T cell one. Humoral immunity is B cell mediated immunity, and acquired immunity is the immunity which we are acquiring during our lifetime, which we are learning, acquiring during our life process. So, the correct answer will be option number 4. Let us move to the next question. Given below are 2 statements. Statement 1, fig fruit is a non-vegetarian fruit. Non-vegetarian fruit it is telling, as it enclosed fig was in it. See, fig and was are in helping each other. What wasp is doing? Wasp is helping in pollination for the fig plant, and fig in turn is providing shelter to wasp for the laying of the eggs. So, fig, wasp and exhibit mutual relationship. Yes, they are having mutual relationship, but they both are helping each other. As they complete its life cycle, without both of them, if they mutually can't help each other, they will not be able to complete their life cycle. And fig fruit gets pollinated by fig wasp. Correct. but statement 1 is incorrect. The crunchy taste of the fig that is due to the seeds, not due to the presence of the vas. So it is not a non-vegetarian fruit. So statement 1 is incorrect, but statement 2 is correct. Option 2 is the correct answer. Let's see what the next question holds for us. Given below are two statements. One is labeled as assertion A and the other is labeled as a reason R. Assertion says what? Cells of the tapetum possess dense cytoplasm and generally have more than one nucleus. Yes, correct. Tapetum is the innermost layer of the anther wall, which is helping in providing nourishment for the pollen grain. And as it is more than one nucleus, it will be having more nourishment. Let's see the reason. Presence of more than one nucleus in the tapetum increases the efficiency of nourishing the developing microscope mother cell. Yes, because of the presence of more than one nucleus, it is helping in the nourishment. That is also correct. And this is in light of the above statement, we have to choose which is the correct option. Both A and R are true. Yes, and R is also the correct explanation for the reassertion. Because of the presence of many nucleus, multinucleate stage, it is helping in the nourishment. So option 3 is the correct answer. Let's see the next question. From the statements given below, choose the correct option. What are the options? Options A says, the eukaryotic ribosomes are 80S and the prokaryotic ribosomes are 70S. What is S? Sydberg units, the sedimentation coefficient. Yes, this statement is true. Let's read the next one. Each ribosome has 2 subunits. Yes, each ribosome is having 2 subunits. The 2 subunits of 80S will be 60S and 40S. We are not going to add them. Yes, it is 60S and 40S. While that of 70S are 50S and 30S. That is also absolutely correct. So we have seen that A, B, C, 3 of the statements are correct. Let's move to the next option. The 2 subunits of 80S ribosomes are 60S and 20S. No, it is 40S. That of 70S and 50S are 50 and 20. No. What it is? It is 30S. This statement is wrong. Option E, the 2 subunits of 80S are 60S and not 30S. It is 40S. And that of 70S are 50S and 30S. That is correct, but here this is wrong. So, which are the correct options? Option A, B and C. Let's see what is the option. A, B and C are true. Third option is the correct answer. Next question says, which of the following enzyme contains heme as the prostatic group? Heme, iron containing group. The prostatic group is a cofactor which is tightly bound. So, which is the heme containing prostatic group? Catalase enzyme. Succinate dehydrogenase will be having FAD and carbonic anhydrase will have zinc, but heme is the prostatic group for the catalase enzyme. The next question. What is the name of the blood vessel that carries deoxygenated blood from the body to the heart in the frog? We are asking that deoxygenated blood from the body. We are not asking from the lungs. So, we will not mark anything which is related to the lungs, but we are talking about from the body to the heart. So, pulmonary means lungs. So, this option automatically rules out. Vena cava, yes, vena cava will bring deoxygenated blood from the body to the heart. Aorta will carry oxygenated blood from the heart to the different parts of the body. So, it is also the wrong option. Pulmonary, again the word, it comes from lungs. So, the correct answer will be vena cava. The second option is the correct option. The next question says, Given below are the stages in the life cycle of pteridophytes. Arrange the following stages in the correct sequence. The body of pteridophyte is diploid, sporophyte body, right? So, in a diploid body, what should undergo? Meiosis, reduction division. The meiosis will take place in the pteridophyte plant body, which is diploid. So, the first step will be meiosis in the spore mother cell. B will be the first thing. What it will form after meiosis? Gametophyte, that is prothallus stage. In the gametophyte, what will be produced? The gametes. So, it will, what are the gametes? Archegonia and antheridia in the gametophyte. So, this will be third. After, when the gametes are formed, archegonia and antheridia, they will be transferred. Anthrozoids will move towards the archegonia, that is the fourth step, and when they move together, they will fertilize. When they are the last here will be fertilization, because there will be fusion of the male and the female gametes. So, this will be the last. Let's see which are the options, which is correct. First thing is meiosis in mother cell, that is B, right? Then prothallus stage, A, these two both are correct. Then the third step is formation of archegonia and antheridia in gametophyte, D. Then what is transfer of anthrozoids and then their fusion. Option 3 is the correct answer. The next question says, the interesting question, the blue and the white selective markers have been developed which differentiate recombinant colonies from non-recombinant colonies on the basis of their ability to produce color in the presence of chromogenic substrate. This question was there in the earlier segment also. Let's read out. Statement 1. The blue colored colonies have DNA insert in the plasmid and they are identified as recombinant colonies. No, this statement is wrong. Why? Because when we are inserting a foreign DNA, we are destroying or inhibiting the action of beta galactosidase enzyme, which is responsible for producing blue color. So, what is going to happen here? The blue colored colonies have DNA insert in the plasmid. No, where the insert is there, they will not produce blue colored colonies. So, this statement is wrong. Statement 2, the colonies without blue color have DNA insert in the plasmid and are identified as recombinant colonies. Yes, this statement is correct. So, statement 1 is incorrect, but statement 2 is correct. The next question says, which of the following microbes is not involved in the preparation of household products? They are asking which is not involved. Aspergillus niger is involved in the production of citric acid, which is on the industrial level. So, this is not involved in the household products. Lactobacillus for curd production, so it is involved in the household. Trichoderma polysporum is responsible for cyclosporin, which is an immunosuppressant used during the organ transplant. So, it is also on the industrial level. Saccharomyces cerevisiae, that is yeast, and Propionibacterium shermani is used for the cheese production, that is also in the household product. So, Aspergillus niger and Trichoderma polysporum A and C are used for the industrial level. So, they are asking not involved in the household one. So, it will be A and C. Option 4 is the correct answer. Let us move on to the next question. Silencing of specific messenger RNA is possible via RNAi, RNA interference, because of. Why RNA interference is taking place? As we know that RNA is single stranded. If we make it double stranded, it will not be doing the process of translation, because RNA is responsible for producing protein. If it becomes double stranded, it will not be doing the further process. So what is the reason which is making it inhibited? Complementary double stranded RNA. Because of the complementary double stranded RNA, it will not be further making any proteins. The option third is a correct answer. Next question. The complex second of mitochondrial electron transport chain, ETS chain, electron transport chain, is also known as succinate dehydrogenase. What is NADH dehydrogenase? That is the complex 1. Then cytochrome C oxidase is the complex 4. Cytochrome BC1 complex is the third complex, and these all complexes are helping in final reduction and only place where the oxygen will be utilized. So here I have completed 15 questions of biology. Now I would like to request Mithil sir to solve few questions of physics. So,
Thank you, ma'am. Is the sound okay? Check, check, check. Yeah, okay. So, thank you, ma'am, for solving the biology questions. I'll be taking up the next questions of physics. We'll solve 15 questions of physics and then we'll go back to botany or biology again. So, coming to physics, the question number 1 is a parallel plate capacitor made of circular plates is being charged such that the surface charge density on its plates is increasing at a constant rate with time. The magnetic field arising due to the displacement current is. I'll solve the question here. They are asking the magnetic field arising due to the displacement current is. The magnetic field, if you remember the chapter of electromagnetic waves, there is a concept called displacement current. Now, what is displacement current? That if there is a capacitor plate, then electric field is generated between it, and when the charge on the capacitor keeps on increasing, so this electric field will also keep on increasing, and as the electric field keeps on increasing, it will create a magnetic field around it everywhere. So there will be magnetic field inside as well. There will be magnetic field inside as well, which will be circular in form, circular in nature, like this. All in all, the electric field, the space between the capacitor plates will act like a current carrying conductor, where the magnetic field will exist outside as well as inside as well. So this is how the figure of magnetic field will be. Let's choose the correct option. The magnetic field arising due to the displacement current is non-zero everywhere with maximum at the imaginary cylindrical surface. 0 between the plates, no, 0 it is not there. 0 at all places, again it's wrong. Constant between the plates and 0 outside the plates, no. It is present everywhere, nowhere is the magnetic field 0. So, we will choose option number 1. Question number 2. The electric dipole with dipole moment this much is aligned with the direction of a uniform electric field of magnitude 4 into 10 power 5. The dipole is then rotated through an angle of 60 degrees with respect to the electric field. The change in the potential energy of the system or the potential energy of the dipole. We know that when you place a dipole in a uniform magnetic field E and the angle between the dipole and the field is theta, the potential energy is given as minus P dot E. That is minus P E cos theta. So initially the angle is 0. Finally the angle is becoming 60 degrees. We have to find the change in potential energy. So change in potential energy is U final minus U initial, that is minus P E cos 60 minus of minus P E cos 0. If you calculate this, you will get P into E by 2, because cos 60 is 1 by 2. So the change in potential energy is P by 2. If you put P, the value of P is 5 into 10 power minus 6. Electric field is 4 into 10 power 5 by 2. So this will give you 10 power minus, sorry, it will give you exactly 1. If you calculate this, you will get exactly 1. So the answer is 1 joule. Coming to the next question, let me just recheck the calculation. This is 2 into 2, find 10, 10 into 10 power 5 is 10 power 6, 10 power 6 into 10 power minus 6 will give you 1. Question number 3, a ball of mass 0.5 kg is dropped from a height of 40 meter. The ball hits the ground and rises to a height of 10 meter. The impulse imparted to the ball during the collision, when the ground collision with the ground is, if this is the ground and this is the ball, it goes down, hits and then rises up to 10 meters. It is dropped at 40 meters. So when it is here, it will have a speed of root 2 g into 40, and when it is rising up, because it is rising only up to 10 meters, its speed will be root of 2 g into 10. This formula comes from V square is equal to 2as. V square minus U square equals 2as. So the change in velocity is root of 2 g into 40 minus root of 2 g into 10. If you put the value of g, root 2 into 10, and inside you will get root 40 minus root 10. This will be the change in velocity. After the change in velocity, now we have to multiply the mass to find the impulse. So impulse will be mass into change in velocity. So that is 0.5 into change in velocity. If you calculate and put all the values with, even if you put g is equal to 10, you will get an approximate answer. That answer will come out to be roughly 7 Newton seconds. So you have to find the change in velocity before hitting and after hitting and multiply it with mass. The intensity of transmitted light when a Polaroid sheet placed between two cross Polaroids is 22.5 degree from the polarization axis. So they are saying there are two sheets, between them you have placed one more sheet. There are total 3 sheets now. From the first sheet, the intensity is I naught. Angle between these two is 22.5. Angle between these two will be 90 minus 22.5. The reason being, they have said that the two plane sheets, Polaroid sheets, were crossed. What does this crossed mean? That this sheet and the last sheet were at an angle of 90 degree. The polarization axis will be at an angle of 90 degree from each other. So between this is 22.5, this one is 90 minus 22.5. Let us check what is the intensity. We know that intensity according to Malus law is given as I naught into cos square 22.5. That will be the intensity which is coming out from here, I. Let's call this I dash, and this final intensity will be I double dash. This I double dash will be equal to I dash into cos square of 90 minus 22.5. So totally this will become I naught into cos square 22.5 into cos 90 minus 22.5 will become sine of 22.5. I will write this as sine of 22.5. So this becomes sine square 22.5. Now what we can do is we can multiply 4 and divide 4, and this will become I naught by 4 into 2 cos 22.5 into sine of 22.5. 2 cos theta sine theta is sine of 2 theta. So that is the formula we will use here, and ultimately the answer will become I double dash is equal to I naught by 4 into sine of 45 degrees, sine square of 45 degrees, this is whole square, and that is 1 by root 2, square of that is 1 by 2, so you will get I naught by 8. So the intensity which is coming out from the last polaroid is I naught by 8. This is how you solve this question. Next question, the kinetic energies of two similar cars A and B are 100 joules and 225 joules respectively. On applying brakes, car A stops after 1000 seconds, B stops after 1500 seconds. I am sorry, it is stopping after 1000 meters and 1500 meters. If Fa and Fb are the forces applied by the brakes on cars A and B respectively, then the ratio Fa by Fb will be what? Now we know that total work done in stopping will be the change in kinetic energy. Since both of them are stopping, they are losing their complete kinetic energy of 100 joules or 25 joules. So the the work done is Fa into the distance, which is 1000, is equal to change in kinetic energy of the first car will be 100. For the second one, it will be Fb. The work done is Fb into 1500 into is equal to 225. If we take the ratio of these two, you will get Fa by Fb. If we cancel out and try to calculate this, this will be 2 by 3. This is 25 into 4, 25 into 9, this is 2, this is 3. So we get Fa by Fb as 2 by 3. So the correct answer is fourth option, which is 2 by 3. Next question. A little complicated question. It looks complicated, but if you are able to get the hint here, you will see that this much section is forming a Wheatstone bridge, and it is a balanced Wheatstone bridge. Let me help you do that by rotating the diagram. Let's see if I can rotate the diagram. Okay, I'll try to rotate it the other way. We have to find the current through the battery. Let us see. Let's look at this question this way. If you look at the points B and E, points B and E, this is E, this is B, and I draw the diagram like this. This is 3 ohms, this is 5 ohms, this is 6 ohms, this is 1.5, and this is 2.5, 3, 5, and this is 6 ohm. If you look at this circuit properly, it is a balanced Wheatstone bridge because the ratio 3 by 1.5 or 5 by 2.5 is constant. So this 6 ohm resistor is actually useless. Because the 6 ohm resistor is useless, we can just choose to remove it and then solve the rest of the circuit. So the rest of the circuit is this 3 and 5 will be in series, so that will be 8. This 1.5 and 2.5 will make it 4. So 8 ohms, 4 ohms, and the idea is if the whole thing, this whole complex looking part of the circuit can be solved, I will rotate it again. If you look at this circuit again, so the whole of this circuit has an equivalent resistance of equivalent of 8 and 4, and that is 8 by 3. So the equivalent resistance of that part is 8 by 3. So I can replace this whole circuit with 8 by 3. So all in all, the circuit looks like this. This is 8 by 3. This is 1.5. This is 1 by 3. This is 5.5. And finally, this is the battery. If we see, all of them are in series. So 8 by 3 plus 1 is 9 by 3, which is 3. 3 plus 1.5 plus 5.5. 1.5 plus 5.5 is 7. 7 plus 3 will give you 10. So the equivalent resistance of the circuit is 10 ohms. So the current through the battery will be V by R, where the voltage is 5 and the resistance is 10. So we get the current as 0.5 ampere. A complex looking question, but if you observe properly, it is a very simple question where we just have to remove the 6 ohm resistor because of Wheatstone bridge being balanced. Next question. Let's see. 3 identical heat conducting rods are connected in series as shown in the figure. The rods on the sides have thermal conductivity 2K, while in the middle has thermal conductivity K. The left end is at 3T temperature, right end is at T temperature. Temperature here is T1, temperature here is T2. We have to find the ratio of T1 and T2. Now to solve this, we have to, let us analyze the question this way. First we will try to find the equivalent thermal resistance. Thermal resistance of the whole thing will be thermal resistance of 1, 2 and 3, that is R1 plus R2 plus R3. R1 plus R2 plus R3. Now what is the thermal resistance of a rod? It is L by Ka. Since all of them are equal in dimensions, identical rods, length and area will be same. The first one will be L by 2K into A, second one will be L by K into A, third one will be L by 2K into A. So L by 2 plus L by 2, 1 by 2 plus 1 by 2 will be 1, 1 plus 1 will be 2. So this will come out to be L2L by Ka. This is the equivalent thermal resistance of the circuit. Not a circuit, it is a thermal circuit. Now let us check. We can say the heat which is flowing through from the first to the second, or heat which is flowing through the first rod and heat which is flowing through the third rod will be same because they are in series. So I am equating the heat current through each one, rate of heat flow through each one. What is the formula of rate of heat flow? Q dot is equal to 2Ka 3T minus T1, because the temperature difference is 3T minus T1, divided by length which is L, and through the last one will be 2K into A into T2 minus T by L, or rather, since we have found the equivalent, let us use that equivalent this way. Through the whole section of 3L length, we can say that the temperature or the Q dot can also be written as temperature difference divided by R, or R equivalent across the whole section. That is, temperature difference across the whole thing will be 3T minus T divided by R equivalent. R equivalent is 2L by Ka. So if you look at this equation, I am comparing the first rod to the whole equivalent rod, and if we start cancelling out the constants or the same things, we will get 2 into 3T minus T1 is equal to 2T by 2. From here we get T1. What will be T1? 3T minus T by 2, that will be 2.5T, that is 5 by 2T. Similarly, if we repeat the same process for the second rod, comparing it with the whole rod, it will come out to be 2K into A into, through the second rod temperature difference is T2 minus T divided by L is equal to, the whole rod will have temperature difference of 2T and equivalent resistance of 2L by Ka. So L and L cancel, K and Ka cancel, this 2 and 2 is also cancelled. We get here T2 is equal to T plus T by 2, which is 3T by 2. Look at T1, look at T2, the ratio of T1 and T2 will come out to be 5 by 3. So the correct answer of this question is 5 by 3. A little bit calculative, but worth it. Next question. I'm sorry. In an oscillating spring mass system, a spring is connected to a box filled with sand. As the box oscillates, sand leaks out slowly out of the box vertically, so that the average frequency omega and average amplitude A of the system change with time t. Okay, there is a box and it is hanging and it is oscillating, and the mass of the box is continuously decreasing because the sand which is filled inside it is constantly being leaking out. Which one of the following options schematically depicts these changes correctly? Okay, let us observe it this way. If we start by looking at omega, which is the angular frequency, omega is given as root k by m. Now k is a constant value because the spring constant is not changing, so omega will only depend on 1 by root of mass. Since mass of the block is decreasing, omega will increase. So we have to choose an option where omega is increasing. So it could be option 1, it could not be 2, it could be 3, or option number 4. It could be 1 or 3 or 4. 2 is eliminated. Now we check what happens to the amplitude. Now I will try to give a non-mathematical way of getting to this answer. Let me try to solve this using just intuition. The mass of the block is reducing, so what will happen to the amplitude of the spring block system? Let us compare it this way. Suppose if the block has a mass m initially when it was filled with, if the block had mass m when it was filled with completely sand, the mass is m, and I release the block such that the spring is at natural length NL. Okay, the spring is at natural length and it is released from here. Then how far can it go? Because we know that this is natural length and this is the equilibrium position. The equilibrium position, we know it is mg by k distance below the natural length of the spring. So here the amplitude becomes mg by k. Suppose after the sand has completely leaked out and the mass of the block now becomes, supposedly it becomes half, that is now the block is half the mass, m by 2, and now you release it from its natural length. Then what will be the new equilibrium position? The new equilibrium position will be mg by 2k, because the mass has become half. The equilibrium distance from the natural length of the spring will also become half. So if you can see, the amplitude has reduced. Amplitude has reduced. If you release a system like this and a system like this from their initial positions, their amplitude of the half block mass will be less. Taking this as our initial and final position of our situation, we can say that amplitude will decrease. Amplitude will decrease. And out of them, which option has amplitude decreasing? We can see option number 4. So option number 4 is the correct answer. Next question. AB is a part of an electrical circuit. The potential difference VA minus VB at the instant when current is 2 ampere and is increasing at a rate of 1 ampere per second is. Now if you see, the current is passing through an inductor, a battery and a resistor. We know that when the current passes from A to B, across the inductor a back EMF is created. The back EMF is given as minus L di by dt. So how much back EMF is created? Minus L. What is L? L value is given as 1 Henry. It is 1 Henry. So 1 into di by dt is also 1, because current is changing at 1 ampere per second. So it is overall, the inductor is producing a back EMF of minus 1 volts. So here a back EMF of minus 1 volts is created, 1 volt. So the potential will decrease when you go from A to this point. Potential will reduce by 1 volt. When you go from here to here, potential will again decrease by 5 volts. Here it is decreasing by 1 volt, And when you hop from year to year, again through a resistor, potential always decreases in the direction of the current. So again, how much potential will decrease? I into R, that is minus current is 2 ampere, resistance is 2, so we get total minus 4. So from A to B, the total potential drop is minus 1, minus 5, minus 4, total 10 volts. So VA minus VB is 10 volts. Coming to the next question, a particle of mass m moving around the origin with a constant force F pulling it towards the origin. There is a particle which is moving with constant force F pulling it towards the origin. If Bohr model is used to describe the motion, the radius r of the nth orbit and the particle speed v in the orbit depends on ns. Let us observe this. We know that this force will provide the centrifugal force. So we write F is equal to mv square by r, and remember that this F is constant. So F equals mv square by r. From here I can say mv square is equal to F into r. Let us keep this aside. Second thing, we know that for a Bohr's model, the angular momentum mvr is equal to nh by 2 pi. What I am going to do is, I am going to find v from here. So, or rather I will find r from here. R will come out to be nh by 2 pi m into v, and place this r in the first equation. So let us see what the first equation gives me. mv square equals F into r, which is nh by 2 pi mv. This will become v cube, and here if you see all the rest of the things are constant, so v cube will be proportional to n. So v will be proportional to n power 1 by 3. So v is proportional to n power 1 by 3. Now I will put this here. So r, I am sorry, I will put that here in the first equation again. Since v is proportional to 1 by 3, in the first equation I will now get m v square is proportional to, from the first equation I can directly write r is proportional to v square, and v is proportional to n power 1 by 3. So r will be proportional to n power 2 by 3, n power 2 by 3. So the correct option for this one will be r is proportional to n power 2 by 3 and v is proportional to n power 1 by 3. Question number 11, in some appropriate units, time t and position x are related as t equals x square plus x. The acceleration of the particle is, this is question number 11. We know that t is given as x square plus x. If we want acceleration from this, let us differentiate this dt by dx. This will give me 2x plus 1. What is dt by dx? This is a reciprocal of velocity. So 1 by v is equal to 2x plus 1. Then v can be written as 1 by 2x plus 1. Now from velocity we want acceleration as a function of x. So acceleration can be written as v dv by dx. V can be written as 1 by 2x plus 1, and differentiation of this will come out to be minus 2 by 2x plus 1 whole square. So the final answer will become minus 2 by 2x plus 1 whole cube. Acceleration is minus 2 by 2x plus 1 whole cube. Option number 4 is correct. Next question, number 12, a model for quantized motion of an electron in a uniform magnetic field B states that the flux passing through the orbit of the electron is nh by e, where n is an integer, h is Planck's constant, and e is magnitude of electron's charge. According to the model, the magnetic moment of an electron in its lowest energy state lowest energy state means n is equal to 1, ground state, right? We have to find the magnetic moment. Let us see. The electron is moving in a uniform magnetic field due to a magnetic field B. So that means this B is providing the centrifugal force. So we can say that QVB, which is the magnetic force on a charge, is equal to mv square by r. Charge is E, so I will write EVB equals mv square by r. I'm sorry. Here V and V can be cancelled, and I can write velocity is EB r by m. Let's keep this relation one side and let's observe. They are asking us to find magnetic moment. Magnetic moment is I into A. Now when an electron is moving in a circular orbit, the current can be written as charge by time period. Charge by time period into area, which is pi r square. So we can write E by 2 pi r by velocity, which is the time period, circumference by speed, into pi r square. Further, if you see, r and r can be cancelled out and we get magnetic moment as E, pi and pi is also cancelled, E by 2 into V into r. Now if you observe very carefully, we can write mass and divide by mass here, so that this numerator will become an angular momentum magnetic moment m can be written as E by 2m into mvr, where mvr is nh by 2 pi. nh by 2 pi. I'm sorry. We are going to use the first relation here. So the first relation will be magnetic moment is equal to E V into pi r square by 2 pi r. Here V can be written as EB by EBR by m. So V can be written as EBR by m into pi r square by 2 pi r and this r and r can be cancelled. I can write this as E square B by 2 pi m into pi r square Also they have given us flux. The flux passing through the orbit is nh by E. So the flux is nh by E is equal to magnetic field into area, which is pi r square. So from here I can write pi r square is equal to nh by EB. If I substitute it here, I get the magnetic moment as E square B by 2 pi m into nh by EB, where n is equal to 1. E and E can be cancelled, B and B can be cancelled. So magnetic moment will come out to be E h by 2 pi m. E h by 2 pi m. That is option number 4. So the question 12 has answer h e by 2 pi m or E h by 2 pi m. Question number 13. A microscope has an objective of focal length 2 cm, eyepiece of focal length 4 cm and the tube length of 40 cm If the distance of distinct vision is 25 centimeters, the magnification of the microscope is, for microscope the magnification is given as 1 plus d by, magnification at distinct vision is given as 25. If you see the relaxed magnification, you will get it as dl by fo into fe. dl by fo into fe, that is the relaxed magnification. What is d given? D is given as 25. Length of the tube is 40 divided by fo, objective focal length is 2 and eyepiece focal length is 4. So, if you calculate this, you will get 250 by 2, which is 125. So, the correct answer is 125 magnification for the compound microscope. Here I believe they should have been more clear and could have told that they are asking the magnification at a relaxed eye, not for the best magnification at distinct vision. Otherwise the formula will be different, answer will be little different and it would not match any of them. Question number 14. There are two inclined surfaces of equal length L and same angle of inclination 45 with the horizontal. One of them is rough, other is perfectly smooth. A given body takes 2 times as much time to slide down a rough surface than the smooth surface. So, there are two surfaces at an angle of 45. This is a rough surface, this is a smooth surface, and the body takes twice the time. Let us say the body takes t time to come down here, then it will take 2t time to come down here. Distance covered in both of them is same. Now we know that when the body is coming down across a smooth surface, the acceleration is g sin theta, where theta is 45, and when it is coming down a rough surface, the acceleration is given as g sin theta minus mu g cos theta. So, we can use the formula S equals ut plus half at square, that is L is equal to half into A is g sin theta minus mu g cos theta. Putting theta as 45 degrees, we get g by root 2 minus mu g by root 2 into t square. I am sorry, the time will be 2t square, 2t whole square. Here in the smooth case, the distance is half into g sin theta, which is g by root 2 into t square. If we take the ratio of both of them, we get all the things will be cancelled out, L and L will get cancelled out, g by root 2 will cancel out, half will also get cancelled out, t square will get cancelled out. We get here, we get 1 minus mu into 4 is equal to 1. So, 1 minus mu is equal to 1 by 4, we get mu is equal to 3 by 4. 3 by 4 is 0.75, we get the coefficient of friction as 0.75. Question number 15. A full wave rectifier circuit with diodes D1 and D2 are shown in the figure. If input supply voltage is 220 in sin 100 pi t, then at t equals 15 milliseconds, what is the biasing of diodes D1 and D2 they are asking. So, let us observe this. If you put t is equal to 15 milliseconds, that is 15 into 10 power minus 3, then the voltage will turn out to be 220 sin 100 pi into 15 by 1000. If you calculate this, this will come out to be 1.5 t, 220 sin 1.5 t can be written, 1.5 pi can be written as 3 pi by 2. We know that sin 3 pi by 2 is minus 1. So, the voltage will be minus 220 volts. If voltage is minus 220 volts, it means that this side will be negative, this side will be positive. So, this side will also be negative, this side will be positive. So, D1 will have a negative voltage here on the P side, on the P side of the diode. So, this will be in reverse bias. and D2, since it is positive on this side, this will be in forward bias. So D1 is reverse bias, D2 is forward bias. The option is D1 is reverse, D2 is forward, option number 2. So option number 2 is the correct answer. So we have solved 15 questions of physics. I would now request our next teacher, Shanaz Madam of Biology, to take up the further discussion of biology questions. Let me welcome Shanaz Madam here. Just give us 2 minutes, we'll keep everything ready. By that time, I request all of you to be ready with your question paper for biology as well, and prepare for the rest of the discussion of the paper. We have completed, I believe, 45 questions of discussion till now. We are going to take further more time. Keep your dinner with you also ready, so that you are not hungry. I know all of you have worked really hard and you have been hungry since, I believe, 1 p.m. today. Have some dinner, keep your notes ready, keep your question paper ready, and be ready for the rest of the question paper analysis. I welcome Shanaz Madam to come to the screen.
Thank you, sir. Hi students, I am Shanaz from the Department of Biology, RVLH. Okay, so we have our first question which is from the zoology part. So in frog, the renal portal system is a special venous connection that acts to link. So whenever we have the word renal, so we understand that we are talking about kidney, right? So whenever you see the word renal, you understand that we are talking about kidney. So in frog, we have a special system that is forming a connection between kidney and lower parts of the body, and that is called as the renal portal system. So the correct option here is 2. So let's move on to the next question. Which of the following are the post-transcriptional events in a eukaryotic cell? So whenever we are talking about a eukaryotic cell, so we know that there are 3 processes that happen after transcription. So what are those? First is splicing, then we have capping, and you also have tailing, right? So these are the 3 post-transcriptional events that happen in the eukaryotic cell. So now let us look at the statements. So statement A is transport of pre-mRNA to the cytoplasm prior to splicing. So this doesn't happen because our mRNA is transported only after all the modifications have been done. So this is false. Removal of introns and joining of exons. Now this process is called as splicing. We know that introns are the interrupted regions which do not code, so we have to remove them. Addition of methyl group at the 5' end of the hnRNA. Now this process is called as capping, right? So this also happens. Now the statement D is addition of adenine residues at the 3' end of hnRNA.
Now this process we know that is capping, sorry, we know that is tailing. So base pairing of two complementary RNAs, we know that in our cells the RNAs are single stranded, they cannot be double stranded, so obviously there is no complementary base pairing in RNAs, so this is false. So now we have to choose what the correct options here. So right, so B, C and D are correct, so we will go with the answer as option 4. Now let's move on to the next question. Polymerase chain reaction amplifies DNA following the equation. Now we know that if you have to amplify your gene or a fragment of DNA, we have a process which is called as PCR. So whenever we are doing PCR, there is an equation that helps us to understand the number of copies that are produced after a certain set of cycles. So that formula is given as 2 to the power of n, where n is nothing but, where n is number of cycles of PCR, right. So the correct option here is 2 to the power of n. So that helps us to calculate the total number of copies of our gene or the fragment of DNA that has been produced after a certain set of, or after a certain set of cycles, right. So the answer is option 4. Let's move on to the next question. So given below are two statements. One is like, so we have an assertion and a reason type of a question here. So both wind and water pollinated flowers are not very colorful. So this is true. So they are not very colorful and they do not produce nectar. Obviously, right. So when wind and water are the pollinators, why do we have to do anything related to attraction? Because you need not attract them. So this is true. The flowers produce enormous amount of pollen grains in wind and water pollinated flowers. So this is also true because we know that the wind and water pollination are more of a chance process. So both the assertion and reason are correct, but reason is nowhere giving an explanation for the assertion. So hence we can say that assertion and reason are true, but reason is not the correct explanation of the assertion. So we will choose the answer as option 4. So now let's move on to the next question. Epiphytes that are growing on a mango branch is an example of which of the following? Now we all know what are epiphytes, right? The plants that grow on other plants are called as epiphytes. Now epiphytes here are an example for commensalism. Now we all know what is commensalism, right? It is an association between two organisms where one organism is benefited and the other is neither benefited nor harmed. Now in this example, the epiphyte is getting benefited and the mango tree is neither getting benefited nor harmed. So we will choose the answer as option 3. Let's move on to the next question. In human pregnancy, the major organ systems are formed at the end of 12 weeks. So this is true. So at the end of 12 weeks means after how many months? After 3 months, right? So this is true. In human pregnancy, the major organ systems are formed at the end of 8 weeks. Now this statement is very contradictory because just now in the statement A you saw that it is formed after 12 weeks. So this is false. In human pregnancy, heart is formed after 1 month of gestation or pregnancy. So this is true. In human pregnancy, limbs and digits develop by the end of 2nd month. So this statement is also true. So in human pregnancy, the appearance of hair is usually observed in the 5th month. So obviously this is also true. So only the statement B is false here. So we have to choose correct answer over here. So we will choose the correct answer as A, C, D and E. So the correct answer is A, C, D and E. So the option is 2, right. So we will move on to the next question. Identify the part of a bioreactor which is used as a foam breaker from the given figure. So now here when you look at these parts, B is the motor, right. B is motor. So this is a bioreactor. So this is simple stirred tank bioreactor which is given here. So B is motor. C is the foam breaker. A is the flat blade impeller, and through D we are going to pass the sterile air. So the question is asking us to choose the foam breaker here. So the answer is 2, C. So moving on to the next question. Frogs respire in water by skin and buccal cavity, and on land by skin, buccal cavity and lungs. So here when you look at the respiration of frog in water, so it can respire only through the skin, but it does not respire through the buccal cavity. So this part is false. And on land it can respire by skin, buccal cavity as well as lungs. So this part is correct. So the correct answer is, the statement is false for water but correct for land. So here the answer is option 1. Let's move on to the next question. Consider the following statements regarding the function of adrenal medullary hormones. Now what are the adrenal medullary hormones? It is nothing but the adrenaline and the noradrenaline, or the epinephrine and the norepinephrine. So what are the functions of these hormones? It causes pupillary dilation. This is false because it causes the pupillary dilation and not constriction. It causes the pupils to dilate and not to constrict. It is a hyperglycemic hormone. It is true, it increases the blood glucose level, so it is a hyperglycemic hormone. It causes piloerection, so this is also true. It increases the strength of heart contraction, this is also true. We know that the adrenal medullary hormones are called as the hormones of flight or emergency hormones. So they do all of this but not the pupillary constriction. So here they are asking to choose something which are the functions of the medullary hormone. So we can choose B, C, D, and hence the correct answer would be option 4. You have to exclude A over here. Let's move on to the next question. Read the following statements on the plant growth and development. So parthenocarpy can be induced by auxins. So this is true. We can induce parthenocarpy by using plant growth hormones like auxins, right? So plant growth regulators can be involved in promotion. So when they are involved in promotion, you call it as plant growth promoters, and when you are having them inhibiting the growth, you call them as plant growth inhibitors, such as your ABA and ethylene. These are inhibitors. Your auxins, gibberellins and cytokinins are plant growth promoters. So dedifferentiation is a prerequisite for redifferentiation. So what does this statement mean? You know that, so once the plant has grown, all the meristematic tissues, most of the meristematic tissues, they lose the property to divide and they differentiate and they become the permanent cells. But at times these permanent cells can again regain the property to divide and become meristematic, right? For example, interfascicular cambium. So these are the meristems that are formed after dedifferentiation, right? So now this process where the permanent cells become meristematic again is called as dedifferentiation. So now this process is very much required, because if your meristematic tissue has to again differentiate into permanent, you call that process as redifferentiation, right? So hope you are understanding. You have permanent tissues which are actually formed from meristematic by differentiation. These permanent tissues again become meristematic by a process called as dedifferentiation. Then meristematic again becomes permanent by a process called as redifferentiation. So if you want redifferentiation to happen, you need dedifferentiation as a prerequisite. So this is true. Abscisic acid is a plant growth promoter. Just now I told you that ABA or abscisic acid is a plant growth inhibitor. So this is false. Apical dominance promotes the growth of lateral buds. When you look at the word apical dominance, obviously it's not going to promote the lateral buds, but it is going to inhibit the growth of the lateral buds. So here you have to choose the option with all correct statements. So how many statements are correct here? A, B and C are correct. So the answer is option 3. Which of the following hormones released from the pituitary is actually synthesized in the hypothalamus? Now we know that our posterior pituitary does not synthesize any hormone but stores and secretes hormones which are ADH, that is antidiuretic hormone, and also oxytocin. So ADH is also called as vasopressin, right? So then who is synthesizing these two hormones if the posterior pituitary is just storing them and releasing? It is the hypothalamus, right? So what is the correct answer? So you have to choose a hormone which is synthesized by the hypothalamus among these. So it is your ADH or the antidiuretic hormone. So the answer is option 4. Let's move on to the next question. Which one of the following is an example of non-distilled alcoholic beverage produced by yeast? Now all of them are produced by yeast, but the only difference is in the way they are processed, whether they are produced after distillation or without distillation. So here the alcoholic beverage which is produced without distillation is beer, and all others are produced by distillation, right? So the correct answer is option 1 over here. Let's move on to the next question. What is the pattern of inheritance for polygenic trait? Now what is a polygenic trait? So a trait or a character which is controlled by more than one gene. For example, human skin color and human height. Here more than one gene are controlling the character. So we clearly know that polygenic inheritance is something that is deviating from the Mendelian principles. It's not following the Mendelian pattern of inheritance. So hence the option we will choose is 4, non-Mendelian inheritance pattern. So here we have a match the following about the sperm. So head, we know that the head carries the genetic material. The head is having the nucleus which is carrying the sperm DNA. So head is genetic material. So A is 4. So we have option 3 and 4 as A. Then the middle piece, what does the middle piece have? It has a lot of mitochondria which is providing energy. So B is 3. So the correct answer is option 3. Now let us look at C. Acrosome has enzymes in it. So C is 1 and tail is helping in the sperm motility. So D is 2. So the correct option here is option 3. Let's move on to the next question. Which of the following is an example of a zygomorphic flower? What is a zygomorphic flower? A flower which cannot be or which can be divided into two equal halves only in one single plane. Such a flower is called as a zygomorphic flower, right? So among these only P is a zygomorphic flower because it belongs to Fabaceae or Leguminosae, and here they have vexillary yeast division, because of which you can divide the flower only in one particular plane into two equal halves. So the correct answer is P. Okay students, so we have solved few questions from biology. So up next we will have our chemistry lecturer solving few MCQs from chemistry. So hold tight, sit back, so till we start with the next chemistry questions.
Good evening, all of you. I'm sure you just finished with biology. Now we are going to start with chemistry, continuing with what we have completed till now. I am Sunita Acharya, the Senior Chemistry Faculty in RV Learning Hub. Let's start with the chemistry questions now. Okay. Which of the following statements are true? The question is asked. Unlike gallium, which has a very high melting point, and cesium has low melting point, we are aware that both gallium and cesium have got low melting point. So this statement is not true. Let's look at question option or B statement. On the Pauling scale, electronegativity values of nitrogen and chlorine are not the same. In fact, they are same. So this statement is also not true. If you look at argon, potassium ion, chloride ion, calcium 2+ and sulfide ion, all of them are isoelectronic species. So this statement is true. The correct order of the first ionization enthalpy of sodium, magnesium, aluminium, you can see very easily that magnesium is having electronic configuration as 1s2, 2s2, 2p6 and 3s2. As it is very difficult to remove an electron from the 3s2 energy level, therefore, the first ionization enthalpy of magnesium is found to be the highest. So, you understand that the correct order of first ionization enthalpy, option D, is also not true here. Now, we go towards the statement given E. They say the atomic radius of cesium is greater than that of lithium and rubidium, and we understand that this again is a true statement. So, if you are going to observe that, you are finding that you will find option A as well as option E is found to be very true, as well as option our C. So, we will pick the answer here as the fourth one. Going forward, let us take the next question. The standard heat of formation of barium 2+ ion. They have given the standard enthalpy of formation of sulfate ion and also the standard heat of crystallization of barium sulfate solid. So, let us write down the equation for the enthalpy of crystallization of barium sulfate. So, it is going to be barium ions along with sulfate ions to produce the crystalline solid barium sulfate. This is the crystalline solid, okay, and the value of the enthalpy of hydration is given to you, and they are telling us that the enthalpy of formation of barium sulfate solid is also given to you as minus 349 kilocalorie, and they want us to find out the enthalpy of formation of barium ion. So, we are having the formula as enthalpy of the crystallization is equal to the enthalpy of products minus enthalpy of our reactants. So, what are our products? That is barium sulfate. You are having the value given for barium sulfate, and you also know the standard heat of formation of sulfate ion. This is our enthalpy of formation of barium ion we need to find, right? And we also have the enthalpy of formation of our sulfate ion, SO4 2- ion, and we are having the values given for the product, which is the value given as minus 4.5, and here it is given as minus 2.16. So, when you are substituting the enthalpy of our crystallization and the value for enthalpy of barium sulfate formation, you are arriving at the answer as option 3, minus 128.5. So, going forward, let us go to the next question. In this particular question, when you are asked to match the pair, it's the easiest one. Let us look at it. Here, they are talking about humidity. The moment you say it is humidity, what you understand, you are able to get it that it is the solubility of liquid in gas. Solubility of liquid in gas. That means A must match with 2. And next you are talking about alloys. What are alloys? Alloys are solid in solid. So you go for this. So immediately when you are able to identify 2 of them, you can immediately pick the answer for our question as option 4, where you can immediately say C is nothing but an amalgam. What is an amalgam? It is liquid in solid. And what is our smoke given to us? It is nothing but solid in gas. So easily you can pick the answer as option 4 here. Going forward, let us look at this question. They are giving us a reaction. Carbon is reacting with hydrogen to give rise to methane gas. Very well. The value of delta H is given. Please observe the value. It is minus 74.8. That means it is which kind of a reaction? It is an exothermic reaction. So now, whenever there is an exothermic reaction, we know for a reaction to be exothermic, we are having the enthalpy of the product to be less than the enthalpy of the reactant. Now let us start observing the graphs. Here the product is less than reactant, but here they are indicating the energy of activation. We don't want that. We want the value of delta H. Here again you are observing the product is higher, so we don't go for this option. Now look at this. Here the product is having lower value than the reactant, and we are definitely getting the value of the delta H. So the answer for this particular question is option 3. Very, very simple, easy to answer such type of questions. Now let us go to the next question. When you are looking at this, it's a jackpot question, very easy. They say a sugar given is X. It is found in honey. It's a keto sugar. The clue is given here, keto sugar. So we are aware that there is one keto hexose with us, and that is fructose. So the answer for this particular question is option D. Going forward, let us go to the next question. The total number of possible isomers, both structural as well as stereoisomers of cyclic ethers having a molecular formula C4H8O. Now let's see how do we go about writing the structural and stereoisomers for cyclic ethers. So 4 carbon atoms, 1 oxygen atom. So definitely I am going to have one structure where we are having 1 oxygen atom and we are having 4 carbon atoms. This is one structural arrangement, and we will have one isomer for this. Okay. Next we could write another structure where we are having structure written like this. So this is 1, 2, 3, 4. We will go for the structure in this manner. We will write, yeah, the structure is this. So now I will observe, we could have this carbon atom. If you will observe, are you having different bonds attached to it? So could it be a chiral carbon atom? Let us keep that in our mind. Okay. So we are having one chiral carbon atom. So you could have 2 isomers here. Okay. Now I could write this formation structure in this manner. There is no chiral carbon atom here. So therefore you will have only one isomer. Okay. I could write probably another structural arrangement in this manner. So we are having 1, 2, 3, 4 carbon atoms with an oxygen. Are you noticing that this carbon atom, which is supposed to have different groups attached to it, so it is chiral. So it could have 2 isomers. I could write the structure like this. Okay. Are you noticing that there are again how many possible isomers? One can be cis, one can be trans, and it can also be a meso compound. So altogether we are arriving at 10 isomers in this particular question as the answer. So going forward, let us go to the next question. For this particular reaction A giving you to B, the backward reaction, the rate constant is higher than that of the forward reaction, and it is by a factor of 2500 at 1000 Kelvin. Okay. So they have given the value of also R. So now what we understand, we know that Kc is equal to K for forward reaction by K for backward reaction. And they say it is the backward reaction is higher than the forward by a factor of 2500. So this is going to be 1 by 2500. Okay. So 1 by 2500, we can keep it like that also, or we can do the simplification, 4 into 10 power minus 3. Fine. Now I find the value of delta N here. Delta N is found to be 1 number of moles of gaseous products minus that of the reactants, and we substitute, we get the formula for Kp. Kp is equal to Kc into RT to the power delta N. So you go on substituting, you know the value of Kc, it is found to be 1 by 2500 into R value you are given, we substitute the value of R 0.0831, 0.0831, temperature is given to you 1000 and to the power 1. So on simplification for this particular question, we are going to get the answer as 0.033. So you will get this as the answer for this particular question. So it's a simple problem, just substitution and simplification. Let's go forward to the next question. When we look at question number 68, the ratio of the wavelengths of the light absorbed by the hydrogen, they have given us the value of N as 2 to 3. So you are supposed to find out the value of nu bar. It is found to be RH into 1 by N1 square minus 1 by N2 square. So if you are going to substitute, it is found to be, the value is 2. So N1 is 2, so it is 2 square minus the value of N2 is 3, so it is 3 square. So you are getting RH into, this is 3 square, 2 square, this is 1 by 4 minus 1 by 9. So this is found to be RH into, this is 9 minus 4 divided by 4 into 9. This is the value. Then you are having the other one, where N is 4 and other value of N2 is found to be 6. So you are finding out the value of nu bar 1, this is nu bar 2. This is RH into 1 by, I substitute directly, this is 4 square minus 1 by 6 square. So you are having RH into 1 by 4 4s are 36, 4 4s are 16, I'm sorry. 4 4s are, it is 16 and 6 6 are 36. So you are having RH into 36 minus 16 by 16 into 36. You are getting the value of nu bar 1 and nu bar 2. but they are talking about the wavelength. So when they talk about the wavelength, it is lambda 1 is the inverse of this. Okay, and lambda 2 will be the inverse of this. And when you are going to take the ratio of lambda 1 by lambda 2, and on substitution, you arrive at the answer as 1 by 4. So, the simplification is there, and just go about it substituting and get your answer for this particular question. Okay, that was question number 68. Let us go further to the next question. They are talking about the molar conductivity lambda m for 0.050 mole per liter solution of a monobasic acid as 90 Simon centimeter square per mole. And they want us to find out the degree of dissociation, that means alpha. And they have given us the lambda naught value of the cation and as well as that of the anion. So, let us find out, applying our Kohlrausch law, you can find out lambda naught m. Lambda naught m will be value of the cation added with that of the anion. So, when you substitute, it is found to be 349.6 plus 50.4. You get the answer as 400. Units are Simon centimeter square per mole. Now you are supposed to find out the degree of dissociation alpha. Degree of dissociation alpha is equal to lambda m by lambda naught m. So the lambda m value is given to you as 90. Here it is given to you as 400. So if you go simplifying, you will get the answer as 0.0225. It's a very simple calculation. So the answer is option 1, because this is 9 by 4, or you can also do it as 9 by 4 into 10 power 1. So this is 4 divided 9 by 4. So 4 times if you do, it will be 2.2, and into 10 power minus 1. So that comes to be, this comes to be how much? 2.2 approximately. We can stop there because we are having 2-3 answers very close. So we will go for 2.2 and into 10 power minus 1. So the answer comes to be 0.225. Straightforward calculation. Okay. So question number 69, the answer is option number 1. Going forward, let us go to the next question. We are having 5 moles of liquid X and 10 moles of liquid Y, and they are making a solution with a vapor pressure of 70 torr. The number of moles of X is given and Y is given. So the number of moles of X is found to be 5, number of moles of Y is found to be 10. And they are talking about the vapor pressures of pure X and Y. So let us find out the mole fraction. The mole fraction for X will be number of moles by total number of moles. Okay, here the mole fraction for Y will be number of moles by total number of moles. Now let us find out the total pressure. If you want to find out P total, so you are going to have 5 mole fraction into the pressure given. Pressure given for X is 63 torr, okay, plus here it is 10 by 15. The pressure given for Y is 78 torr. Let us substitute and we simplify and we get the answer approximately as 73. So the calculated value is 73 total pressure and the given value is 70. As the calculated value is greater than the given value, it is having a negative deviation. So, the solution is showing a negative deviation because the calculated value is much higher than the one which is given to you. So, the answer is option 4. Clear? So, let's go further for the next question. Question number 71. Okay. For this particular question, we have to find out equal number of atoms. So, it's very simple. Let us talk about A. What is it? It is 212 divided by molar mass. It is how many times? It is almost 2 times. Correct. Now, how many atoms are there? Let us check. This is 2 moles, right? And how many atoms are there? 2 atoms of sodium, 1 atom of carbon, so 3, and 3 oxygen atoms. So, altogether how many? 6. So, 2 into 6, we have 12 atoms. Let's talk about B. B, it is 248 mass by molar mass given to you as 62. You get it as 4. Okay. Let us check the number of atoms. 2 atoms of sodium and 1 atom of oxygen. So, there are altogether how many? 3. So, how many atoms? 12 atoms. Okay. Let's come to C. What is it? It is mass 240 divided by molar mass 40. So, it is how many times? 6. And now, let us check the number of atoms. 1, 2, 3. So, 6 into 3. How many? 18. Okay. Let's go to D. This is very easy. It is 12 grams of hydrogen by 2. So, it's going to be how many? 6. How many atoms are there? 2. So, it is found to be, multiplied by 2, it's found to be 12 atoms. Let's go to E. What is it? 220 divided by 44. 220 divided by 44. So, it is approximately, I think so, yeah, we get it as 5. And we are having 1, 2, 3 atoms. So, total 15. Now, let us check which of the following have equal number of atoms. It is A, then B, and you have D. So, it is A, B, and D. Option 4. It's very simple. Stoichiometry question. So, let's go forward to the next question. Yeah. Which of the following are paramagnetic? The trick is very easy here. Please identify which of the ligands are weak field ligands and which are strong field. Wherever you are having weak field ligands, they are paramagnetic. Whichever are strong field ligands, they are diamagnetic. So, we know that chlorine is a weak ligand. Again, carbon monoxide is found to be a strong ligand. Cyanide, strong ligand. So, diamagnetic, H2O, a weak ligand, and triphenylphosphine, a strong ligand. So, which are the weak field ligands? These are the two. So, what are these two able to show paramagnetic property? So, A and D could be the ones which are paramagnetic. So, it should be option 1. Very easily you can solve such questions. So, let us go for the next question. If the half-life of a first order reaction is 1 minute, then the time required for 99.9% completion of the reaction is closest to. Again, we are aware of a simple formula. T 99.9% is 10 times T 50%. And the value for half-life is given as how many minutes? 1 minute. So, what is the answer? 10 minutes. So, the answer should be option 2. So, it is a very easy, straightforward problem. Let us go to the next question. We are having energy and radius of the first Bohr orbit of helium ion and lithium ion to be calculated. So, we will see in all the options, they have given first the energy of lithium and the radius, then helium is coming. So, first we will find out the energy for lithium and the radius for lithium ion, then we will go for the helium ion. So, let us see what's the formula. The formula for the energy is found to be minus RH into what we have, Z square by N square. Okay, and the formula for the radius is taken as A naught into what is that? N square by Z. Fine. So, if you will start substituting for lithium ion, Li2+. So, what is the atomic number of lithium? It is found to be 3. Fine. So, what is this value? E is equal to minus the RH value is given as 2.18 into the value of Z is 3 square. So, 3 square is 9 by N is 1 because it has lost 2 electrons. So, that is 9 by 1. So, 2.18 into 9, you can immediately start checking out the answer when you have already given this, you have come to this stage, alright. So, when we are going towards the radius, when you are calculating the value for the radius, the value of A is 52.9, we can take it as 53 into the value of N is of course 1 and Z is found to be, here the value is found to be 3. So, 52.9 divided by 3. Easily you can start identifying the answer. I have already given the substituted values in this particular for lithium ion. I am sure you have started calculating already. I will go forward for helium. In the meanwhile, you can report your answer. When you go for our helium, it is going to be minus 2.18 into the value of Z for helium. For helium, He plus. What is the value of Z? It is found to be 2. So, it is found to be, here we will take it as 2 square by 1. Okay. Fine. And the radius calculation is 52.9 into 1 by 2. So, when you are already able to substitute all the values and you will start calculating very easily, you could account or you can calculate the answer, and the answer for the question will be a option which will tally. You can see this 2.18 into 9. So, it should come approximately 19.62 into 10 power minus 18, and here it will be 17.6, 52.3 divided by 9, 52.9 by 3. So, it should be option 3. Very easily you can get the values after simplification. So, that was the answer for question number 74. Let us go for the next question. 75th question. Among the given compounds 1 to 3, the correct order of bond dissociation energy of the CH bond, which is marked with an asterisk. Very well. Let us look at this. There is little ambiguity, because what are they trying to ask? Whether it is the dissociation to give you formation of a free radical. Generally, we are going to go thinking about the formation of a free radical because of the bond dissociation of the CH bond. So, or whether it is resulting in the loss of H plus ion or H minus. Let's see that. I am sure we all are aware that in benzene, the carbon atom is sp2 hybridized, and the sp2 hybridized carbon hydrogen bond is not easy to break. The bond is shorter and stronger, difficult to break. Now, when you come towards compound 2, in compound 2, I want you to observe that the carbon-carbon triple bond is there. So, it is an sp carbon. So, sp carbon, the bond is still shorter, more difficult to break. So, compound again 2 is very difficult to break the carbon-hydrogen bond. Okay. Now, when you go towards the compound 3, what happens in case of compound 3? This is allylic. This is an allylic hydrogen. And we are aware that the allylic carbon-hydrogen bonds are very easily broken. Very, very easily broken. As a result, it is very easiest to break. So, the bond dissociation energy is definitely going to be the lowest. So, it is found that you can have the bond breaking of benzene hydrogen to be very difficult. Then you are having here sp carbon. This is little better, little easier. Then we come to the option or compound 3. So, you could pick the answer as 4, but it is an ambiguous question. So, it is yet not very, very clear what kind of bond cleavage they are looking forward. So, these were all the 75th question which we have finished in the NEET section for chemistry. Thank you, everyone.
Hello everyone, I am Tejaswini, Biology Faculty, RVLH. Let us solve some more questions from question number 151 to 165. Okay. The question here is, while trying to find out the characteristic of a newly found animal, a researcher did the histology of adult animal and observed a cavity with presence of mesodermal tissue towards the body wall, but no mesodermal tissue was observed towards the alimentary canal. Now, if you look at the words here, the cavity is referring to coelom, and here the body wall has a mesodermal lining, but it is not present towards the alimentary canal. So, such a coelom wherein it is not lined by mesodermal layer is known as pseudocoelom. So, here directly you can make out that the animal in which they studied the histology of the adult animal, when they saw the cavity, it is not lined by mesodermal tissue entirely. So, such an animal is pseudocoelomate. So, there is no confusion here. So, we just have to look into these keywords which is given in the data. Let us see the next one. Now, it is a statement problem. Let us see the statement here. Statement 1, in the floral formula, the symbol stands for zygomorphic. Now, it is a direct question. This symbol is not zygomorphic, it is actinomorphic. So, statement 1 is wrong. So, statement 2, in the floral formula, the symbol stands for actinomorphic nature of the flower, and this symbol stands for superior ovary. So, this is correct. Now, in the light of the above statement, choose the correct answer. So, we have to choose the correct statement here. Now, if you look at the statement, statement 1 is wrong because it is not zygomorphic, whereas statement 2 is correct. So, statement 1 is incorrect, statement 2 is correct. So, the correct option here is option 2. Now, next is also a statement problem. Statement 1, in ecosystem, there is unidirectional flow of energy of sun from producers to consumer. Now, it is a directly NCERT statement. So, it is unidirectional from producer to consumer. So, the first statement is correct. Second statement, ecosystems are exempted from second law of thermodynamics. Now, here ecosystems are not exempted from second law of thermodynamics, but here they have given as they are exempted. So, second statement is wrong. Now, in the light of the above statement, choose the appropriate answer. So, statement 1 is correct, statement 2 is incorrect. So, option 1 is your answer. Now, again a direct question here, which of the following is the unit of productivity of an ecosystem? Now, they are directly asking about the unit here. Either it can be given in grams per meter square per year, but here it is given in the cals energy. So, directly you can say kilocals meter square per year. So, this is the option, second option is your answer. Now, next question is a pedigree chart. With the help of the given pedigree chart, find the probability of the birth of a child having no disease and being a carrier. So, they want you to find out the probability of a child who is not having a disease and a carrier which is having one allele of the gene wherein it is a recessive condition, and they are specifically talking about the F3 generation. Now, if you look at the pedigree chart here, they have given 3 generations. Now, F1, if you look at the F1 here, now she is a carrier mother. Carrier mother has 2 children, one is daughter and one is son, and son is affected. So, a carrier mother giving rise to a affected son is possible in X-linked recessive disease like color blindness or hemophilia. Now, in the F2 generation, if you look, you can see that the affected male is married to a carrier woman. Now, observe the F3 generation. Now, since she is a carrier, now he is affected, the carrier female will have an affected X chromosome, whereas affected male is having a X chromosome which is having the mutated gene or the recessive gene. Now, when they produce the offspring which is seen in the F3 generation, there is a possibility of one daughter who will be a carrier, one daughter who will be affected, and son, one who will be affected, and another son who will be normal. So now they want you to tell the probability of a child having no disease but being a carrier. Now this is seen that one of the daughter will be a carrier. So directly you can say that it is about 25%, which is given as 1 by 4. So option 3 is your answer. Now next question, in the seeds of cereals, the outer covering of endosperm separates the embryo by a protein rich layer. Now this is again a direct question. You just have to recall the structure of the monocot seed and try to recall what is the layer which separates the embryo and the endosperm. So since it is a protein rich layer, directly you can go in for aleurone layer. Now next is match the following. So chlorophyll A, B, xanthophyll and carotenoids. You have already studied these photosynthetic pigments, and by paper chromatographic method you can isolate these pigments. So we can identify the color here. So chlorophyll A is blue green color. So A is matched with 3. So 2 option directly you can eliminate. So A is matched with 3 in the other 2 option. Let us see for chlorophyll B. Chlorophyll B is yellow green in color. So B is matched with 1. So only 4th option is having the option here. Let us check with the others. Xanthophyll is yellow in color. So C is matched with 2. Carotenoid is yellow to yellow, orange in color, D 4. So your answer is option 4. Now next is again a direct question. Who proposed that genetic code for amino acid should be made up of 3 nucleotides? So this was proposed by George Gamow. So it is directly based on your knowledge, memory based question, recall, and you can identify the answer. Your option is 3. Again a direct question. Histones are enriched with, so histones are the proteins which are combined with DNA to form the chromatin. So if you recall the amino acids present in histones, histones are enriched with lysine and arginine. So option 3 is the correct answer. Now next question, which of the following enzymes are not essential for gene cloning? Now gene cloning is nothing but you have studied the process in recombinant DNA technology. If you recall the enzymes required in recombinant DNA technology, we know that we require restriction enzymes to cut the DNA. DNA ligase is required to join the DNA. DNA polymerase is required for amplifying the DNA, whereas DNA mutase and DNA recombinase is the enzymes we do not require in gene cloning or recombinant DNA technology. So C and D is not required. So the option which is C and D only is your option 3. So your answer is option 3. So the next one, a specialized membranous structure in a prokaryotic cell which helps in cell wall formation, DNA replication and respiration. Again, if you have recalled the structure of the prokaryotic cell, you know that in bacteria they have these cellular, the cell membranous structure which are the foldings which helps in respiration, cell wall formation, DNA replication, which is known as mesosome. So cristae, endoplasmic reticulum, chromatophores are the pigment containing membranous foldings. cristae present in mitochondria. Endoplasmic reticulum is a cellular cell organelle present in an eukaryote. So, hence the remaining option is wrong. The only option which is suitable is option 3, mesosome. Now, another direct question, which factor is important for termination of transcription? Now, you have studied the factors responsible for initiation as well as termination. Sigma is an initiation factor, whereas rho is an termination factor. So, directly you can go in for your option 1, rho, which is termination factor responsible for termination of transcription. Which of the following statement is correct about location of the male frog copulatory pad? Again a direct question. If you recall the morphology of frog, if you see the morphological features of a male frog, you can recall that the copulatory pad is present in the first digit of the forelimb. So, very direct question it is. So, your option is 2. Next is a diagram based question based on your structure of the nephron. Now, let us see the question. Which of the following diagrams is correct with regard to proximal, which is labeled as P, and distal, which is labeled as D, part of the nephron? So, that is they are referring to PCT and DCT. Now, the arrow mark indicates the selective reabsorption process as well as tubular secretion process. So, if you see the arrow mark here, the arrow mark going out is nothing but it is indicating selective reabsorption. So, what are the components that is selectively reabsorbed in PCT as well as DCT? And the inward arrow mark basically talking about tubular secretion process, which are the components that is secreted into PCT and DCT. So, it is a diagram based question. So, the diagram is, you can always recall the diagram from the NCERT textbook. So, here you can see that the options given here talks about the tubular secretion and selective reabsorption. So, here the first option is talking about PCT correctly, whereas NaCl salt is not being tubular secretion. During tubular secretion, NaCl is not secreted out. Okay. So, same way here if you see, so the water, which is very important here, so it is secreted into the PCT, so it is wrong. Okay. Again here water is shown which is being secreted in tubular secretion. So, third option is also wrong, whereas the fourth option shows it correctly. So, during tubular secretion, hydrogen and ammonium is secreted into PCT, whereas bicarbonates, salt and water is being reabsorbed. From DCT, salt, water and bicarbonates are reabsorbed, whereas potassium and hydrogen ion is secreted. So, the fourth option shows the correct representation of the PCT and the DCT. So, hence your answer is option 4. Okay. Now, let us see the last question here. Identify the statement that is not correct. So, we are choosing the incorrect statement. Going with the first statement, antigen binding site is located at C-terminal region. Now, recall the structure of the antibody molecule. So, in the antibody, the antigen binding site is located at the N-terminal, but not the C-terminal. So, the first statement itself is not correct. Let us see other statement. Constant region of heavy and light chains are located in C-terminus, that is correct. Each antibody has 2 light chains and heavy chains, that is correct. The heavy and light chains are held together by disulfide bonds. Remaining statements are correct. The incorrect statement here is option 1. So, this is the questions from 151 to 165. I will hand over the stage to Marvin sir to continue with physics.
Thank you, ma'am. Hello, students. So, you had a very enriching session till now. I think you should grab a cup of water and probably sit again for the next set of questions that we are going to discuss. Once again, welcome to RV Learning Hub. Today, we are going to discuss some questions from physics. I will be discussing another 15 set of questions, students. You know that NEET exam is one of the most toughest exams that you can see because of the number of students who take a number of seats. But you know what, irrespective of how that paper toughness is, only the best students usually get the best rank. So, this year is one such examination where though the questions have come towards the medium and the hard part, it is still the best students who will get the rank. So, do not worry students, fasten your seat belts and just see how well you have done your physics part. So, let me give a quick introduction. I am Marvin, I teach physics, I am in RVPU South with 12 years of experience. I am going to discuss the next question now. So, we have a uniform rod of mass 20 kg which is of length 5 meter, which is kept against a vertical wall. It is a standard question. The angle is made as 60 degree is what they have claimed with respect to the vertical. So, this angle here is going to be 30 degree. Dear students, whenever we get questions like this, we have to do rotational as well as translational equilibrium. Looking at the contact forces, we have one force here, one force here, the weight that is acting, which is mg. So, totally there are 3 forces which are there. Along with that, the question also says there is frictional force, and that frictional force is what we have to find, right. Now, entire system should be in equilibrium. Let me call this as N1, this as N2 and this as mg and this as the frictional force F. So, upward equal to downward, left equal to right, that is how you equate the forces at equilibrium. From this we can write that N1 can be equated to the frictional force. Now, how do we write frictional force as? It is going to be mu times of N2, because at the point of contact N2 becomes a normal reaction. How do we find it? Now, let us go to the next part, which is rotational equilibrium. Considering this point as the axis of rotation, you notice that N1 is trying to make the rod rotate in anti-clockwise direction, mg is trying to make it rotate in clockwise direction, whereas this is acting at the point of rotation. So, therefore, it does not produce any rotation. On that context, if we have to find out the torque applied by N1, torque is going to be force into perpendicular distance. They have given the length here as 5 meter, right. If this is 30 degree, the perpendicular distance from the axis of rotation, which is here, is going to be 5 cos 30, and the force is going to be N1. So, what is the torque due to N1? The torque due to N1 is going to be N1 into 5 cos 30. Now, similarly, if you take torque due to mg, mg is acting in downward direction, right. So, this is going to be a clockwise torque. Look at the perpendicular distance from the axis of rotation, this is the perpendicular distance. Now, this is half of the length. So, we can write this as 5 by 2, the perpendicular distance is going to be the sine component. Therefore, can we say here in the next case torque is going to be due to mg, and that is going to be mg into 5 by 2 becomes the length, and the component is going to be sine 30 degree. So, from this we get one more equation. Dear students, on solving this part, we are going to get the value of N2, and then we are going to find out the frictional force, which is nothing but N1 itself. So, directly if I find N1 from here, we get the answer. Equating both of them, you are going to get N1 in this particular case. Here cos 30 is root 3 by 2, you will get a root 3 dependence there, and on simplifying mass here, which is going to be 20 kg, g is 10, which is 200, and we have a root 3 by 2 dependence here. On solving that, we will get it as answer 100 root 3. Going to the next question, students. So, we have two identical charge conducting spheres. This is based on Coulomb's law. You must have done questions on this in various cases. Let us see how to do it. So, if there are two conducting spheres, they are, let us say, having charge q and q. First, they are brought in contact. So, each of them will attain a charge of q by 2 and q by 2. Later, they are bringing a third sphere which is uncharged, again brought in contact with the first sphere. Now, the charge on this is 0, charge on this is going to be q by 2, right. So, when B is finally removed from both of them, we need to find out what will be the new force of attraction. So, the second case, they are bringing in contact the sphere which is first brought in contact in A, then each of them became q by 2 and q by 2, and after that the third identical uncharged conductor is brought in contact with sphere A, they are telling. So, this is going to be brought in contact with the second sphere. So, what is the charge on this sphere now? Q by 2, but what is the charge on this sphere here? This is q. So, when they try to interact with each other, the net charge is going to become q plus q by 2. So, what is the net charge? It is 3 q by 2. Now, that is going to get divided into 2 halves. So, what is charge on each sphere then? It will be 3 q by 4. Got it? So, in the end, the charge on the sphere here is 3 q by 4, whereas the charge on this sphere is q by 2. Coulomb's law we know, F is proportional to the product of the charges. In the first case, it is proportional to q square. In the second case, it is proportional to the product of the new charges, which is q by 2 as well as 3 q by 4. On looking at the ratios, one can easily make out that the force is going to be 3 F by 8 times the original force. Let us move on to the next one. There are two cities X and Y which is connected by a regular bus service. This is actually a question from NCERT. In the new NCERT textbook, this question has been deleted, but this was a question very much part of the old NCERT textbook. Okay, let us see the question. So, they are leaving, the bus is leaving at regular intervals both from X as well as Y. Now, let us say that these are the positions of the buses which are regularly left at intervals t. There is one more person who is cycling, and that person is driving a scooter with a speed of 60 km per hour. Therefore, if you consider the velocity of this person with respect to the moving bus, that means the moving bus velocity will be treated as 0. So, with respect to the moving bus, if you see, the velocity of this person will be appearing to be going in backward direction, because the bus is going in forward direction, this person will appear to go in backward direction. So, what will be the relative velocity of this person with respect to the bus? That is going to be v minus the speed is given as 60. Now, let us say that the distance here between the two buses is d, relative distance. So, for the person to travel from here to here, that is from one bus to another bus, they have given the time as how much? 30 minutes. That means, we can also write time as half hour. So, this is velocity, time is half hour, this is going to be the distance between the two buses when the person is seeing the bus going in the same direction. Now, the same thing if I reverse it, the relative velocities will get added because these two are moving in opposite direction, then the equation becomes v plus 60. Now, what is the time? They have to go to 10 minutes, 10 is nothing but 1 by 6 hours, and the distance between the two buses is still going to be the same because they are going with same speed, which is given as v, let us say. So, the distance is going to be the same. Now, equating both of them, because left hand sides, right hand sides are equal, left hand side should be equal. Equating both of this, if you solve, dear students, you will get the speed as 120 km per hour. Next, we have to find out the time interval. How to find the time interval? You have the distance which can be calculated from this equation. If you put v as 120, we get 120 minus 60 divided by 2 as distance, right. So, 60 by 2 distance is going to be 30 kilometers. Now, you know distance, and you also have to find the time. You know the speed of the bus, right. The speed of the bus is 120. So, can we just say that for the bus, speed is equal to distance by time, right, where speed is going to be 120, distance we have got it as 30, and we need to find out time. So, time will be 30 by 120 in hours, which is going to be 15 in minutes, making it option 2. Moving on to the next question, students. We have containers with 2 chambers, the volumes are given and they are separated by a partition with thermal insulation, no exchange of heat. Moles is given, pressure is given. We need to find out what happens when the partition is removed. Dear students, whenever we remove a partition, remember it is free expansion, and for free expansion the heat of the first system and the heat of the second system will give the total heat. On that logic, we can actually say that F by 2 mu 1 R1 T1, that will be the total energy of the first, plus F by 2, because they have not mentioned the type of gas, I am taking it as F only, mu 2 R is the universal gas constant, right. So, R T2, that will be equal to the total energy of the system, which is again F by 2. What is the number of moles after removing the partition? Mu 1 plus mu 2, right. R into T becomes a common temperature. Now, if you try and simplify this, students, F by 2 is going to get cancelled. Common temperature therefore can be written as mu R T1 is P1 V1, mu R T2 can be written as P2 V2, divided by number of moles will be mu 1 plus mu 2. From this we will get the common temperature of the system. If you substitute values, we have 1 into the volume is given as 2 plus 2 into volume is given as 3 divided by number of moles is 5 plus 4. On simplifying this, we will get 8 by 9 as a temperature. Now, we need to find out when they have attained equilibrium, what is the final pressure of the system. So, how do we do that part? We can add number of moles, right, that is another concept, where mu 1 plus mu 2 can be written as mu. Now, with this particular concept, if you apply your ideal gas equation, then you are going to get the total pressure of the system as P, the total volume of the system as V1 plus V2 after remove the partition. The number of moles in the system is mu 1 plus mu 2 after removing the partition, and R will be the gas constant, and we have the temperature as 8 by 9. In fact, we have missed the R dependence here, which will come in the denominator. So, V1 and V2 we already know, students, V1 is 2, V2 is 3, mu 1 and mu 2 we already know, which is 5 and 4. On simplifying this, we will get the value of P, and the value of P turns out to be 1.6. You can do the calculations and cross check the same. Moving on to the next one, we need to find out de Broglie wavelength of electron orbiting at n is equal to 2 state. Dear students, in this chapter we would have seen towards the last part they would have shown a connection between dual nature and atoms. What does it say? It says that the integral multiple NH by 2 pi will be equal to MVR. Are we aware of this? Very much? Yeah. This is our postulate of Bohr, right. Okay. Now, how do we find de Broglie wavelength? H by MV is de Broglie wavelength, right, which is 2 pi R by N. R can be written as R naught into N square, N being the principal quantum number. Therefore, you get 2 pi N into R naught, where N here is 2. Substitute as 2, R naught value they have given, students, 0.052. Upon calculation, you will get this answer as 0.67 nanometer. Can check it out, 3 pi can be taken as 3.14. Okay. Moving on to the next one, there is an AC power supply, they have given 220, simple question, frequency is given, resistance, reactance are given for capacitor, inductor. We need to find out current and the phase angle. These are the things that we have to do. How do we do that? Very simple, right? We have everything here. So, Z can be written as root of R square. What is R here? 20 square into XL minus XC the whole square. 25 minus 45 will give you 20. Does not matter if you take plus or minus, because in the end we are looking at square. So, how much is Z, students? 20 root 2 ohms. Now, we have to find out current. We have voltage. Formula simple. Current is V by Z, right, where V is 220, Z we have got it as 20 root 2. Students, a simple calculation here, it is 11 by root 2, 1 by root 2 value is 0.707, right, standard value 11 into 0.707. If you try to do, you will get answer close to 7.8. For finding phase angle, you can use a formula cos phi is equal to R by Z. I think R is given as 20, Z we have got it as 20 root 2, am I right? So, we will get this as 20 by 20 root 2. Once again it is simple, where cos phi is 1 by root 2, meaning what is phi, students? 45 degree, and that makes it as answer 4, 7.8 and 45 degrees. Okay. Let us move on. Next question. A simple question once again, these graphs are standard there in your NCERT textbook, am I right? Photoelectric effect says that the photo current and intensity are directly proportional. So, this is 100% correct, but intensity has nothing to do with frequency. Therefore, photo current and frequency cannot be related. They are independent of each other. This is something that you must have learned. Frequency only affects the energy and the stopping potential, not the intensity or the photo current. So, this is also wrong, this is also wrong, right, because they do not depend on each other. It is not a constant. So, there is only one correct answer there, that is option 3. Only A is correct. Photo current and intensity are directly proportional. Moving on, students, we have the question with oscillations. We have open pipe and closed pipe. So, you can all check the answer key in the description now and cross check with your score, students. Okay? Let us continue. So, we have a question here. A pipe is open at both ends. What is the fundamental frequency for this part? We all know the formula. Fundamental frequency for open pipe is V by 2L. Now, they are telling that to this pipe, half is filled with water. That means, the length which is available for it is only half of the length, and this acts like what pipe? Closed pipe. What is the formula, students? It is V by 4L, but the oscillating length is only 1/2 of the original length. So, if you simplify here, we get closed also as V by 2L, this is V by 2L, that means the frequency remains the same, answer is 4, right. Moving on, next question, we have two identical point masses P and Q suspended by two separate massless strings. So, I think we have to relate it with maximum speed. Do you know the relation, students? Yes, V is equal to A omega is a relation. Now, they want to find out AQ by AP. Let us be very careful here, AQ by AP, not AP by AQ, AQ by AP, which means AQ by AP becomes VQ by VP into, because we are bringing it to the denominator, omega P by omega Q. Now, let us observe one more thing, maximum speeds are same. So, this part gets cancelled, simple. How is omega related to K, students? Omega is equal to root of K by M, right. So, it is going to be P by Q, and that matches with second option, root of K1 by K2. So, AQ by AP will be KP by KQ, KP by KQ meaning the first by the second, got it, simple, root K1 by K2. Let us move on to the next question. We have the output to be found out here. Dear students, here we do not have to apply Boolean algebra, you need not find the equation, you know there are many shortcuts to do this question. The best method to solve it is by substitution. How do we do that? Let us take some example. We will say this is 0, 0 and 0, right. 0 and 0 OR gate would have given you a 0, complement of you would have given a 1. 0, 0, product is 0, complement has given you a 1. 1 and 1 has given you an output of 1 because it is an AND gate, multiplication, simple. So, for inputs of 0 and 0, what did we get output as 1? So, 0, 0, 1 we got, just write it. Now, let us redo this same thing for another possibility which is 0 and 1. So, if you take 0 and 1, this is 0, right students, 0, 1, OR gate meaning addition, 1, complement 0, 0, 1 AND gate which is 0, complement 1. Now, 0 into 1 will give you 0, therefore, for 1 and 0 or 0 and 1, we will get it as 0, right. Move on to the next one which is going to be 1 and 1, this is 1, this is 1, this is also 1. 1 and 1 is going to give you a 1, complement will give you 0, 1 and 1 is 1, complement will give you 0, 0, 0, 0. And what kind of gate is this looking like? Yes, it is a complement of NOR, or it is a complement of OR gate which is called NOR gate. So, answer will be option 2. Moving on, students, this is a little lengthier in calculation, I agree, but we have to do these kind of questions, probably not during the first 2.5 hours, but try these type of questions only towards the end. Also see here, they are telling nearly equal to, where they are not expecting you to do the exact calculation, go by some approximations. Let us see how to solve this. There is volume given, there is number of moles given, and they are telling that some oxygen is withdrawn from the cylinder. I want all of you to observe this term gauge pressure. What does it mean? It is atmospheric pressure plus the given pressure, and they have given the temperature. Now, when you pull out some mass from the system, we are going to observe that the temperature is going to remain same, because the entire process is done at 27. Also, the volume is going to remain same. So, two parameters here are constants. One is volume, other is temperature. Using this idea, we can go for our ideal gas equation, and then we can solve this problem. Let us see how to do that part. First thing, in the initial case, let pressure be P naught, volume is 30 liters. So, I have to go with a conversion 10 power minus 3. Number of moles, we will take it as 18.20 for the first case. R, I will take it as per the question, 100 by 12. And temperature is 300 Kelvin, right? Because they have told 27 degrees Celsius. Wonderful. Let us write one more equation. For the next case, the pressure we will take it as P, and that pressure is 11 atmospheric pressure for gauge. That means the total pressure is going to be 12 atmospheric pressure. Am I right? So, it is 12 into 1.01 into 10 power 5, because we have a conversion also to be done from atmosphere to SI unit. Let us move on. Volume is 30 into 10 power minus 3. You will see a lot of similarities. Mu is there, 100 by 12 is there, 300 is there. Dear students, if you take a ratio, you will see many terms are getting cancelled, right? Let us make it a little easy. Though this is a lengthy calculation, see how I am going to solve it. P naught by 12 into 1.01 into 10 power 5, and we have a 30 here into 10 power minus 3. Got it? Because of volume conversion. This entire thing is going to get cancelled. So, why should I write that? You just have to write the conversion factor, 1.01 into 10 power 5. This 30 got cancelled. 10 by 100 by 12, 300, all these are getting cancelled. What is left out on right hand side, students? We have 18.20 divided by mu. Got it? So, this is going to be the equation where the pressure is P naught, and the pressure after the removal of oxygen is 12 into 1.01 into 10 power 5. Got it? Now, how do we express P naught as? That we can take from the above equation. What is P naught, students? P naught can be written as 18.20 into 100 by 12 into 300 divided by 30 into 10 power minus 3. If I substitute it here, again many terms are going to get cancelled. I will be able to find out what is mu value. So, if you simplify this, students, you are going to get the mu value here, and the number of moles, if you perform the calculation, you will get it as roughly around 3.65 moles. Got it? So, what did we understand from here is that from this entire idea, we got the number of moles that was left behind, which is going to be the difference between the original number of moles and the number of moles after removing, and that delta mu, I am sorry, is 3.65, not the number of moles. In fact, if you do calculations here, you will get the number of moles upon mathematical simplification as roughly 14.56 students. So, 18.20 was initial number of moles, 14.56 is final. Difference we will get it as 3.65. After this, it is very easy, because they have also given the molecular weight. So, how do you find the mass? Simple, right? Mass is going to be number of moles into molecular weight. Remember, this is in grams. You have to convert to kgs. Reason? Options are all in kgs. If you do the calculation here, students, 3.65 into 32 into 10 power minus 3, upon solving this, we will get the answer as 0.116 kg, roughly. You can do rough calculations and get to the answer. Got it? Let us move on to the next question. In a certain camera, we have combinations. These are very simple questions, no students? Yes. So, if we have 4 lenses like this, 4 lenses are put together, what will be the effective power? 1 by f equivalent is 1 by f1 plus 1 by f2, am I right? So on and so forth. But we want powers. How do we write power? P1 plus P2 plus P3 You know what is the beauty of this question? All are P. So 4P is definitely correct. Apart from that, how to find magnification? I am sure you must have learned this in compound microscope concept. If we have magnification of first as ME and other as MO, the image of the first will become object of the second, the object is going to form another image. So, very simple students, total magnification will be the product of individual magnification. With that logic, can we say, because there are 4 of them, the total magnification is m into m into m into m, which is m power 4. So, we have m power 4 as magnification, 4p as the power, making the answer as option 1. Let us move on to the next question. There are 2 gases A and B which are fit. Again a question on the case of kinetic theory of gases. You can see a lot of questions this year on this particular topic. So, let us see how to solve this particular part. So, we have the radius as RA for one of the cylinder, the radius as RB for another cylinder, and both are filled with ideal gases. That is what they say. They have moving under constant pressure, displacements are given to you. Now, if the change in internal energy is same, first thing we have to notice, second thing we have to notice is both are given equal amount of heat. So, heat is there, internal energy is there. What thought comes to your mind, students? Wonderful. Delta Q is equal to delta U plus delta W. That is what you get, right? Where these two are constants, meaning obviously work done also should be the same. Now, how to find work done under constant pressure? Where is that word constant pressure? Yes, I find it here, constant pressure. What is work done under constant pressure? Work done under constant pressure is pressure into V2 minus V1, correct? This is a cylinder, students. How to find volume? We are all experts in mathematics, so I am sure you know about it. Volume can be written as pi r square h, right? So, for the first case, it is pressure into pi into radius of the first one, we will take it as RA. The change or displacement for the first case is given as 16. Now, the same thing is done for the next one, where work done will be the same. Pressure is P, area is pi RB square. And how much is the displacement made? Height, it is 9. Now, because work done is also equal according to first law of thermodynamics, delta Q same, delta U same, work done should be same. Yes, simple, you substitute, you will get the answer, students. We have RA square into 16 as RB square into 9. They want a ratio between RA and RB. So, if you take RA by RB, you are going to get the answer as square root of 9 by 16, which turns out to be 3 by 4. That is the correct answer. Moving on to the next question. We have a balloon and so many things are there, students. Yeah, you know, there is a tendency that you tend to forget sometimes that the question is actually easy. You know why? Let me show you the trick. See here, any equation to be correct must be dimensionally correct. Whether or not it is physically correct, it should be dimensionally correct. So, let us use this idea for solving the problem. Yes, they have given a detailed explanation of what are each terms and other things, but these are common to us. This is time period, the surface tension, area, density and radius. So, just use dimensional analysis maybe to solve these type of questions, where we have time period, we have surface tension, right. Time period is related with surface tension, area, and then we have density and we have radius, right. Let us do the idea, simple. So, time period is m0, L0, T1, right. Surface tension, mass per unit length is going to be m0, L0, T minus 2, force per unit length, I am sorry, force per unit length, area L square, density m1 L power minus 3, radius is L power 1. So, these are given as power alpha, beta, gamma and delta, right. That is what they say, question is clear. Now, what are the powers of m? If you see, we have m power 0 here, we have m power gamma, next L power 0, L power 2b, 2 beta I mean, then we have minus 3 times gamma and we have delta, right. And time, if you see, we have only one dependence, which is minus 2 alpha, all right. I think you have got the answer. You are all very fast. I know, students, you are all very fast, faster than the teacher sometimes. You are right. So, you have to just equate, you get the answer. you are going to see that minus 2 alpha is going to be equal to 1, then alpha is going to be minus 1 by 2. Got it? And then we have, I think I have missed out the gamma dependence here somewhere. You have to check for gamma. Let us go back to the question. We have rho power gamma here, and we have already got alpha as minus 1 by 2, which is matching with this key here and this key here. And then we have to check out for gamma. I think we will get alpha plus gamma as 0. I must have missed it somewhere. Yeah, I get it. I have missed it here. Right, I am doing it too fast, I think. Anyways, you are all catching up very fast, so I do not have to worry. You must have already corrected that. So, we would have got it as gamma plus alpha is 0. Gamma plus alpha is 0 in the sense, what is gamma, students? Gamma is going to be 1 by 2. So, alpha we got minus 1 by 2, gamma we have got 1 by 2. That is perfectly matching here, gamma and alpha, gamma and alpha. So, it has to be 1 or 4, 1 or 4, 2 and 3 ruled out. Next, what do? We cannot solve now. Do you know why? Because we have got here 4 variables, but only 3 equations. Then you are in a stuck situation in exam hall. Do not worry, students, that is why options are given. Do substitution and get the answer. How do we do that? Just equate the power of L, very easy, 2 beta minus 3 gamma plus delta should be 0, is that right? Now, let us see how to proceed further. We have alpha here, we have gamma here, gamma is 1 by 2. So, 2 beta minus 3 by 2 plus delta will be 0, correct, and we will take the values of beta from the options. Let us take first option, because first and foremost let us take first, beta is minus 1. If you put beta as minus 1, you are getting minus 2 minus 3 by 2 plus delta is 0, that means delta is going to be 7 by 2, and that is only there in the first option. Good that you tried it with the first option, you got the first, otherwise it is 4, so simple, right. So, we are going to go with answer of the first option, which is here 7 by 2 is what we have got. All right. We come to the next question, vertical circular motion, a very commonly asked type of question, but let us give the importance to the details. Do not worry about the options, okay. Let us observe the question. Here we have length as L, speed as V naught. How do we solve vertical circular motion? 2 equations, yes, one for energy, one for the FBD. So, let us go with the same strategy. Do not memorize any of the formulas, go with the strategy. Here kinetic energy is half m V naught square at the lowest point, potential energy is 0. What about velocity here? That is what we have to find, V. So, what is kinetic energy? Half m V square. What about the height? See students, here it is L, here it is L opposite. So, L sin theta, then the height is going to be L plus L sin theta. So, m g L plus L sin theta. So, we got one equation from energy. Yeah. Next, what we have to do? We have to apply FBD. Good. If you apply FBD at the lowest point, what do we get? We have mg acting downward, we have some tension acting upward, and the resultant will be acting upward. I am not worried about this. Do you know why? Because I want velocity at P. So, let me take it out and now find it out for P. At P, if you have to find, they are telling that it is getting just slacked at that point. Tension is 0, makes your job easy, no tension. There is going to be component of weight acting along the direction of circular motion, that is equated to centripetal force. So, students, mv square by L, centripetal force at this point is going to be the component of weight acting in this direction. So, we have mg here, this is theta, this is 90 minus theta, this is mg cos 90 minus theta, which is mg sin theta, and thus we have got another equation. After this, I am not going to do much. It is easy for you to understand, students. Take sin theta, substitute here, simplify. I am sure you are going to get the answer here as sin theta by 2 plus 3 sin theta to the power 1 by 2. All right. Now, coming back to one more question that we had done, we had discussed about one of the answers. I think let us look into that question. Once again, we have the small calculations to be changed. I think we are in chemistry and then comes physics. All right, I think I've got it here. All right. Anyways, I think it was the third question that was there. I will give the data of the question. We had a body of mass 0.5 kg students, and it was at a height 40 meters, and then after hitting it came back to a height of 10 meters. We had to find the impulse imparted. We just had to do a small correction there with the signs. If you see, the velocity here is downward, and after hitting it goes upward. So, change in momentum is going to be mv minus of minus mv. So, there we had done an addition, but subtraction I think we had to take a minus sign here, we missed. So, it is going to be 2 times, where velocities are going to be v1 and v2. If you do the free fall concept, v is root of 2 gh, substitute that, it is going to be root of 2 g h1 minus root of 2 g h2. h1 and h2 values are there, which is 40 and 10. They have clearly told you to take it as 9.8. So, this is going to give you the impulse. Minus of minus here is plus. I think that is what we missed last time. On simplifying this, you is going to get the answer as 21 Newton second. You can do with the value, students. 0.5 is mass, h is given, g value 9.8, and you will be able to get the answer. So, this was question number 3 of what we have discussed before. So, next I would like to welcome Sushmita ma'am to take over for the upcoming questions. Thank you, students.
Hello children, this is Sushmita Ghosh, Department of Biology. Stay tuned, we will be back in 2 minutes. Hello all, this is Sushmita Ghosh, Department of Biology. I am going to start with the discussion of biology questions. So, let us start with the first question, question number 121. The question is, which of the following organisms cannot fix nitrogen? The options are Azotobacter, Oscillatoria, Anabina, Volvox, Nostoc. Azotobacter is the free living nitrogen fixing bacteria. Obviously, it fixes nitrogen. Oscillatoria, Anabina and Nostoc, these 3 are cyanobacteria. They have a specialized cell called as heterocyst. Because of that, they can fix nitrogen. So, the organism which cannot fix nitrogen is Volvox, because Volvox is an eukaryotic algae. It is a colonial algae which cannot fix nitrogen. Option is 4, D only. Let us move on to the next question. Which of the following is an example of ex situ conservation? Children, we have already studied about conservation, which is divided into 2, in situ and ex situ. Among that, ex situ conservation is a type of conservation where the organisms are taken out from their natural environment and been protected in a particular area. Okay, like zoological park, even the botanical garden, these are the example. So, the correct option is option 1, zoos and botanical garden. Protected areas, national park, wildlife sanctuaries, these are the example of in situ conservation. Okay. So, let us move on to the next question. Question number 123. Who is known as the father of ecology in India? This is a direct question. Obviously, in India, the father of ecology is Ramdev Mishra. Let us move on to the next. Question number 124. Given below are the 2 statements. Statement 1 is saying, in the RNA world, the RNA is considered the first genetic material evolved to carry out essential life processes. This sentence is to some extent correct. Yes, because RNA is obviously involved in different metabolic processes like splicing, okay, translation, etc. This first sentence is correct. RNA acts as a genetic material. Yes, for some viruses, RNA acts like a genetic material. That is correct.
also acts as a catalyst for some important biochemical reactions in living systems. Yes, the catalyst, one of the example where RNA acts like a catalyst is ribozyme. Being reactive, RNA is unstable. This statement is also correct. RNA is really unstable because RNA is having uracil instead of thymine, and in the pentose sugar, second carbon position is occupied by one hydroxyl group, which makes the RNA easily degradable. So, statement 1 completely is correct. Statement 2, DNA evolved from RNA and is a more stable genetic material. Obviously, DNA is the most stable genetic material. Its double helical strands, being complementary, resist changes by evolving repairing mechanism. Yes, this statement is also correct. So, the correct option is statement 1 and statement 2, which is both the statement 1 and statement 2 are correct. Option 3. Let's move forward to the next question. Question number 125. Given below are two statements. Statement 1 is saying transfer RNA and ribosomal RNA do not interact with mRNA. Is it like that? Children, we know that mRNA is the one on which protein synthesis takes place, and protein synthesis takes place with the help of amino acid. Who brings the amino acid to the site of protein synthesis? That is transfer RNA. And until or unless ribosomal RNA gets associated, okay, the protein synthesis will not begin. So this statement is not at all correct, because RNA, transfer RNA and ribosomal RNA and mRNA together we need for protein synthesis. So they would be interacting with each other. So statement 1 is wrong. Come to the statement 2. RNA interference takes place in all eukaryotic organisms as a method of cellular defense. RNA interference, in the name only meaning is there. Here what is happening? No, with the help of double-stranded RNA, with the formation of double-stranded RNA, somewhere the translation process is being disrupted. Okay, it is a natural process, the mechanism which interferes the translation process. So it is called as RNA interference. So this process is applicable in biotechnological process in controlling the pest. Okay, so statement 2 is actually correct. Statement 1 is wrong. Statement 2 is correct. So statement 1 is incorrect, but statement 2 is correct. Option 2 is the correct option. Let's move forward to the next question. Match the list 1 with the list 2. Heart, erythropoietin. Here the name of the organs and here the name of the hormones what they secrete. So we know that heart actually helps in the secretion of atrial natriuretic factor. This factor is present in the atrial wall. The hormone is getting secreted from atrial wall and it helps to reduce the blood pressure. That is the function. Come to kidney. Kidney is responsible in the secretion of erythropoietin. Erythropoietin is the hormone which is secreted from the kidney, which helps in erythropoiesis, formation of RBC from bone marrow. Okay, so this is actually the function of erythropoietin. Gastrointestinal tract is connected with the secretin. Secretin is a type of hormone which controls the pH of duodenum by secreting the bicarbonate salt from the pancreas, and it also helps to reduce the secretion of gastric juices. Okay, that is the function. Adrenal cortex is related to aldosterone. Aldosterone is the hormone which helps to maintain the blood pressure as well as it helps to maintain the sodium and potassium balance in the body. So the correct option is A is equal to, A is related to the 3, A3, so option 2 it should be. B is equal to 1, okay. C, gastrointestinal tract is related to secretin, C is equal to 4, D is equal to 2. So option 2 is the correct option. Let's move forward. Question number 127. All living members of the class Cyclostomata are. Cyclostomata is the primitive form of vertebrate. Its mouth is circular. That is the name this name has given as Cyclostomata. When it is in adult stage, it lives over the surface of fishes, okay, and feed on fish body only. So they are parasite. As they are living on the surface of fish, so they are ectoparasite. So the correct option is ectoparasite. Okay. Streptokinase produced by bacterium Streptococcus is used for. Streptokinase is the name of the enzyme which is produced from the bacteria called Streptococcus. Streptokinase is an enzyme which helps to remove the blood clot. Actually what happens, it acts over the plasminogen, convert it into plasmin, then plasmin dissolves the fibrin. Fibrin is an important ingredient of blood clot. So this streptokinase is being used for those patients who are suffering from heart attack due to blood clot formation. Understood? So the correct option is it helps in removing the clots from the blood vessels. Question number 129. Role of the water vascular system in echinoderms is, echinodermata, the phylum is recognized because of its water vascular system. You know that this water vascular system is actually everything for them. They do not have proper organ system. So all the functions like digestion, excretion, even the respiration, locomotion, all these processes would be maintained by water vascular system of echinodermata. So the correct option is A, respiration and locomotion, and E, digestion and excretion. That is the one, A and E. Sorry, it should be 1 minute. Excretion and locomotion, respiration and locomotion, and that is the correct option. Excretion, capture and transport of food, digestion and respiration, digestion and excretion. B and C, no. Excretion and locomotion, sorry, is not the correct option. So it should be A and B. Option 3. So this is not a correct option. Match list 1 with the list 2. Pteridophyta, Bryophyta, Angiosperm and Gymnosperm, and this side we have given Salvia, Ginkgo, Molytricum and Salvinia. Pteridophyta is connected with Salvinia. Okay. Bryophyta is a polytrichum. Example of Bryophyta is polytrichum. Example of Angiosperm is Salvia, and example of Gymnosperm is Ginkgo. So the option A is actually 4. A is actually 4. Bryophyta is polytrichum. Angiosperm is Salvia. Angiosperm is C1. A. Pteridophyta is Salvinia. Pteridophyta is Salvinia. Bryophyta should be the polytrichum. Angiosperm is Salvia and Gymnosperm is Ginkgo. So A is connected with 4. 4. Bryophyta should be polytrichum. Bryophyta should be polytrichum. Angiosperm should be 1. C should be 1 and D should be Gymnosperm should be Ginkgo. So option 4 is the correct option. Okay. Question number 131, which are correct? Computed tomography and magnetic resonance imaging. Computed tomography means CT scan. Magnetic resonance imaging is called as MRI. Okay. These detect cancers of internal organs. Yes, these two are the method which can detect the internal organs cancer. Chemotherapy, so this option is correct. Chemotherapeutic drugs are used to kill the non-cancerous cells. No, chemotherapeutic drugs are used to kill the cancerous cells. So this should be wrong option. Alpha interferon activates the cancer patient's immune system and helps in destroying the tumor. Alpha interferon is a type of biological response modifier. What is biological response modifier? These are some protein which are secreted from the virus infected cell, and they are used to protect the healthy cells from the infection of virus. Especially for the cancer treatment, this process is very much involved. Okay, so alpha interferon activates the cancer patient's immune system and helps in destroying the tumor. This statement is correct. Chemotherapeutic drugs are biological response modifiers. No, chemotherapeutic drugs are not. It's alpha interferon, which are a group which belongs to a cytokine protein. So this is a wrong statement. In the case of leukemia, blood cell counts are decreased. It is also a wrong statement because cancer treatment for leukemia, so blood cell count will be increased, especially WBC cell count would be increased. So the option A and C correct. Option A and C, that is 2, second option. What are the potential drawbacks in adoption of the IVF method, in vitro fertilization? You know that nowadays many couples are going for this process, IVF technique, where when they are suffering from infertility, in that case they will adopt this process, in vitro fertilization. So what are the drawbacks of this method? Let us see. High fatality risk to mother. No, there is no risk for mother by adopting this process. So this is not a correct option. Expensive instruments and reagents. Yes, the process is very expensive. This is correct. Husband or wife necessary for being donors. This is not a drawback. This is not a correct option. Less adoption of orphans. Yes, those who are adopting this process, they don't have to go for adoption process. So this is also correct. Not available in India. In India, it is very much available. So this is a wrong statement. Possibility that the early embryo does not survive. Yes, it happens in many cases. Actually, the success rate is less. Okay, so this is also a correct option. So B, D and F, these 3 are correct. This is the correct option. Option 3. Let's move on to the next question. Question number 134. In bryophytes, the gamete help in which one of the following? Okay, gamete. This one is very much important for asexual reproduction in case of liverworts. If you talk about the liverworts, for them the asexual reproduction takes place with the help of that multicellular bud which is present located in the receptacle, which is called as gamete cup. Okay, so gamete actually helps in asexual reproduction. Option 4 is the correct option. Come to question number 135. Given below are 2 statements. One is labeled as assertion A and the other one is labeled as reason R. Assertion. The primary function of the Golgi apparatus is to package the materials made by the endoplasmic reticulum and deliver it to intracellular targets and outside the cell. Children, we have studied about cells topic, and there we have studied that endoplasmic reticulum, Golgi body, these are acting like a factory, like how the things gets manufactured. Okay, so primary ingredients would be given to the factory and final product would be developed. Okay, so endoplasmic reticulum, Golgi body also behave like a factory. So what happens now here? So Golgi apparatus is to package the materials made by the endoplasmic reticulum. This statement is correct. Okay, and deliver it to the intracellular targets and outside the cell. This statement is correct. Reason. Vesicles containing materials made by the endoplasmic reticulum fuse with the cis phase. You know that in the Golgi apparatus there are 2 phases. One is forming phase, cis phase, where the materials are getting formed. Once the formation is done, the final outcome would be delivered through the transfer phase. So there are 2 phases. So obviously vesicles containing materials made by the endoplasmic reticulum fuse with the cis phase, means forming phase of the Golgi apparatus, and they are modified. Golgi body will modify them and release them from the transfer phase. Transfer phase means maturation phase. So this statement is also correct. Okay, so both the statements are true, A and R is the correct explanation of A. So the option 3 is the correct option. Okay, this is all about biology from my side. Now I request Mahesh sir to proceed with the chemistry question answer solving.
Hello everyone, I am Mahesh DR, Senior Lecturer, RUPU College South. I am here to discuss the last 15 questions of NEET exam 2025. So let's begin the discussion. The first question is, Dalton's atomic theory could not explain which of the following? We know that Dalton's atomic theory is in the structure of atom chapter. So it can explain law of multiple proportion, and it can explain law of conservation of mass, and it can explain the law of constant proportion, but it cannot explain the law of gaseous volume, so which is explained by Avogadro's law. So that's why, so the correct option for this is option 2. So let's move on to the next question. The next question is, identify the correct orders against the property mentioned here. The first one, about dipole moment. So the H2O dipole moment is 1.85 d by, next NH3 is 1.45 and HCHCl3 is 1.01. So that's therefore, so dipole moment of H2O is greater than dipole moment of NH3, and which is greater than dipole moment of chloroform. So the correct this is the correct property which is mentioned. Next one, number of lone pair of electrons around each atom. So if you take this xenon, the first one is XeF4. In case of XeF4, so Xe has 8 electrons, 8 plus 4 will be 12, 12 divided by 2 will be 6. That means around xenon in XeF4 there are 6 electron pairs, out of which 4 are bond pairs and 2 lone pairs. That means the first one has 2 lone pairs. Next, XeO3 is 8 plus 0 because oxygen is monovalent atom. So 8 divided by 2 will be equal to 4. Here there are 3 oxygen atoms, so therefore 3 bond pairs and 1 lone pair. So this has 1 lone pair and the previous one has 2 lone pairs. And XeF2, so coming to the XeF2, so Xe is 8 plus 2 which will be equal to 10, 10 divided by 2 will be equal to 5. So therefore 5 electron pairs are there, out of which 2 are bond pairs and 4 are lone pairs. That means it has 4 lone pairs. So this is not the correct order, since XeF4 has only 2 and XeF2 has 4 lone pairs. So then this is not the one. Next, this is about the bond length, OH, CH and NO. So here if you take this bond length, so NO bond length is greater than CH and least bond length is for OH. So this is also not the correct order. The next one is bond enthalpy. You know, it's very simple. So nitrogen to nitrogen triple bond will be there. So triple bond energy is more when compared to oxygen double bond and hydrogen to hydrogen single bond. Hence this one is also correct. So then the correct option for this question is the one which has option A and D, which is nothing but correct option is option 3. So that's what about question number 77. So we moving on to the next question. So now match the list 1 with list 2. So in the list 1 they have given vitamins, and the list 2 they have given the deficiency disease. Now let's try to go one by one. The first one, vitamin B12. We know that vitamin B12 will cause pernicious anemia. So therefore for A it is 4. Next one, vitamin D, we know very well it is rickets. So for B it is 3. Next one, vitamin B2. So vitamin B2 will cause chelosis. So therefore vitamin B2 is given in the C. For C the correct one is 1. And vitamin B6 will cause, the deficiency of vitamin B6 will cause convulsions. So then for D it is 2. Then the correct option will be for this is A4, B3, C1 and D2. A4, B3, C1 and D2. So hence the correct option for this is option 4. So this is again a memory based question here. Moving on to the next one, that is question number 79. The correct order of decreasing basic strength of the given amines is. Look at this. So here they have given 4 amines. The first one is N-ethyl ethanamine. So N-ethyl ethanamine is C2H5 NH C2H5, which is a secondary aliphatic amine, right? So followed by ethanamine. So ethanamine is C2H5 NH2. We know that among aliphatic amines, the secondary amine is more basic when compared to primary amine. Therefore, among these two, N-ethyl ethanamine is more basic, followed by ethanamine. Next 2 amines are aromatic amines. The first one is N-methyl aniline. The N-methyl aniline is this one. So NHCH3. This is again a secondary amine. So secondary amine again is more basic here, since this is attached, this N is attached to a electron donating CH3 group. And the next one is aniline. So you know, so aniline is an aromatic primary amine which is less basic than the secondary aromatic amine. Hence, the most basic one is alkyl amines, and less basic will be aromatic amines. So based on that, so secondary aliphatic amine is more basic, followed by primary amine, followed by secondary aromatic amine, and followed by the primary aromatic amine. Hence, the correct option for the question number 79 is option 1. So moving on to the next question. So the next question is from coordination compounds. The question is, the correct order of wavelength of light absorbed by the following complexes is. So the wavelength of the light absorbed depends upon the strength of the ligand, depends upon strength of ligand and the number of ligands. Strong field ligands will absorb light of lower wavelength and higher frequency. They have asked here, so correct order of wavelength of the light, right? So if you take this Cn and NH3 and H2O, Cn is the strong field ligand, so it causes maximum splitting, hence it absorbs a light of lower wavelength. Then the correct option should contain B as the first one. So here 1 and 2 cannot be the correct options. So since the C is the first option, next either should be 3 or 4. Next, so B, after that Cn, the next strong field ligand is NH3, then it should be A. So B less than A. They absorb light of lower wavelength and higher frequency. Coming to the next one, C and D. So both of them water as a ligand, but here option D contains 6 water molecules as ligand, but it has only 4 water molecules as ligand. In this, the splitting will be more since it is an octahedral field. So D will absorb next lower wavelength and the next wavelength will be for C. So then the correct option is the option which has B, A, D, C. So hence the correct option is option 4. So that's what about the question number 80. So we will move on to the next question, that is question number 81. Again a very simple question. So which one of the following compound does not decolorize bromine water? We know that bromine water will add on to the alkenes or alkynes, or it add on to aniline and phenol. Here this will bromine, so bromine will be adding on to this alkene. So as a result of this, the bromine gets consumed in the reaction, hence the color of the bromine get discharged. The reaction takes place like this. So this is the one, CH double bond CH2 plus Br2, right. So since bromine gets consumed in this reaction, its color will be disappearing. So then you are going to get this compound. So CH, Br, single bond, CH2 Br. That means the first one will decolorize the bromine water. Next one, aniline. You know that aniline reacts with bromine water, resulting in the formation of a bromo derivative. So hence this will also decolorize bromine water. Next, this is a cycloalkane. This cycloalkane doesn't have any unsaturation, that is, it doesn't have any double bond, hence it does not react with bromine water, hence bromine water does not get decolorized. Hence the correct option is option 3. And also if you take phenol, phenol reacts with bromine water, resulting in the formation of 2,4,6 tribromophenol. So question number 81, the correct option is option 3. The next question. So the next question is, predict the major product P in the following sequence of reactions. They have given a cycloalkene which is attached to a methyl group. So they have added this HBr. This is the compound which is given there. To this they have added HBr in presence of benzoyl peroxide. That means addition of HBr takes place against Markovnikov's rule. So where you are going to get Br attached to this carbon atom. So Br, then you are going to get CH3 here and H will be added here. So this is what about the first product by reaction with HBr. Next they have treated that with this product with KCN. When this is treated with KCN, nucleophilic substitution takes place. So resulting in the formation of a cyano compound, which is nothing but CN here, right. Next, in the third step, what they have done is they have used a reducing agent. They reduce this compound using sodium mercury amalgam in presence of ethanol. So where it will undergo reduction to give a primary amine. So you are going to get CH2 NH2, which is the primary amine. Hence the correct option for this question number 82 is option 3. So moving on to the next question, that is question number 83. So again is a match the following type of questions. The question is based on the first year purification techniques, right. So they have given the first one, mixture they have given, and method of separation. You know, CHCl3 and C6H5NH2. So chloroform and alanine have lot difference in their boiling point. Almost the chloroform has a boiling point of 61 degrees Celsius and aniline has a boiling point of about 184 degrees Celsius. Now we know that since there is a lot difference in their boiling point, we can separate these two by simple distillation. That means A, the correct option, the correct matching answer is fourth one. Next one, B. B, they have given crude oil in petroleum industry. So crude oil in petroleum industry can be separated or purified by fractional distillation. That means B, the correct option is option 3. Next one, glycerol from spent lye. We know that glycerol will decompose at its boiling point. That's why it has to be purified by reduced pressure distillation. Distillation and reduced pressure. For C, it is 1. And last one, aniline and water. We know aniline and water can be separated by steam distillation. That is steam distillation. For D, it is 2. So therefore, the option which contains this is A4, B3, C1 and D2. Hence the correct option is option 3 for question number 83. Moving on to the next question. So the next question is, which of the following electronic configuration belong to main group elements? This is a chapter from classification of elements and periodicity in property. We know that the main group elements are S block elements and P block elements. The S block and P block elements are called as main group elements. So whether the element is S block or P block can be decided based on the electronic configuration. So if the last electron enters into S orbital, it is S block element. Yes, here in the option A, the last electron is entering into 3s orbital, hence it is S block element. Whereas in the option B, So it's a d block element because the electron will enter 4s first and then it will enter 3d. So it is nothing but anti-penultimate shell. So that's why this is not a main group element, this is a d block element. Coming to the option C, it is a 4d10, 5s2 and 5p5. Yes, so here also, here the last electron enters into s orbital, hence it is a s block, a main group element. Last electron enters into p block, hence it is a p block element, and which is a main group element. The next one, this is d block element again, and this one is again so d block f block element. So that's why this is not the one. Hence the correct option should be the one which has A and C, that is option 4. Question number 84, the correct option is option 4. Moving on to the next question, that is question number 85. The question is, which one of the following compound can exist as cis and trans isomers? We know that alkenes and cyclic compounds can exhibit geometrical isomerism, that is cis and trans isomers. Let's go one by one. So 1,1 dimethyl cyclopropane. So this is 1,1 dimethyl cyclopropane. So CH3 and CH3 and here H and H. So it cannot exhibit the geometrical isomerism since this carbon has same atom, there is nothing but hydrogen. So hence one is not possible. Then the second one is 1,2 dimethyl cyclohexane. So we know that 1,2 dimethyl cyclohexane is this one. So this is if you take this as 1 and this is 2, this is 1,2 dimethyl cyclohexane, CH3 and CH3, here H and H. This is a trans isomer, and we can also assist isomer, right? This is some isomer of this, which is nothing but CH3, CH3, H and H. Hence, the one which exists as cis trans isomer is so option 2, which is nothing but 1,2 dimethyl cyclohexane, and remaining 2 cannot exhibit geometrical isomerism, hence they cannot form cis and trans isomers. Moving on to the next question, there is question number 86. This question is from equilibrium chapter. The question is phosphoric acid. So phosphoric acid is H3PO4. Phosphoric acid ionizes in 3 steps with their ionization constant values Ka1, Ka2 and Ka3 respectively, while K is the overall ionization constant. So which of the following statements are true? We know that for a acid which has more than one proton, which will undergo in multiple steps, so the overall stability constant is given by, overall ionization constant is given by K is equal to K1 into Ka1 into Ka2 into Ka3. If you take log on both the sides, then it will become log K is equal to log Ka1 plus log Ka2 plus log Ka3. Hence, the first option is correct. Next, H3PO4 is a stronger acid than H2PO4 2 minus and HPO4 2 minus. Yes, definitely. So as this H3PO4 loses one proton, it becomes H2PO4 minus, which is not ready to lose the second proton readily, and same as in case of HPO4 2 minus. Hence this is also correct, because H3PO4 can produce higher concentration of H plus compared to H2PO4 minus and HPO4 2 minus. The next one, we know that the strength of the acids is directly proportional to dissociation constant. You know H3PO4 is a stronger acid, hence it has higher value of Ka. Therefore, Ka1 is greater than Ka2 and greater than Ka3. Now let's go through the option. So before going to D, so is there any option which has all the A, B, C and D? So no, there is no option which is having all the A, B, C and D. Hence the correct option should be the one which contains A, B, C, that is option 2. For question number 86, the correct option is option 2. The next question. So again, this is based on salt analysis. that is about the basic radical, you have to find out the group. So coming to this, let's move on from, in order to solve this question, let's start from B and D, then we'll go through the remaining. Since we have this magnesium and aluminum, so it will be easy for us to get the answer. Now, so it is B. If you take this B, B is magnesium. So magnesium always comes in 6 group basic radical. The 6th group is in 4th option, B is equal to 4. Next, aluminum. So aluminum is a 3rd group basic radical. The aluminum is D. D is group 3 basic radical, which is nothing but 2. Now let's go through the options given here. So you can see here in these 4 option 1, 2, 3, 4, the only one option, that is 4th option, has D is equal to 2. That means aluminum is present in group 3. So hence we can finally and easily say the correct option is option 4, right. So and B is 4, so B is 4 and A is 3. A is nothing but CO2 plus, which is nothing but a group 4 radical, and C is 1. So C is nothing but Pp2 plus, which is a group 1 basic radical. So after finishing the question number 87, we will move on to question number 88. Question number 88 is again based on Lee-Chatelier principle from chapter chemical equilibrium. The question says, asks here, higher yield of NO in N2 plus O2, N2 is in gaseous state, O2 is also in gaseous state, giving rise to 2 moles of NO, which is also in gaseous state, can be obtained at, that means higher yield of NO. How to increase the yield of nitric oxide? Since we know that this is an endothermic reaction, we know that endothermic reaction will be favored by the increase in temperature. That means higher temperature will favor the forward reaction, that is formation of nitric oxide. That's correct. Next, they have given lower temperature. We know that lower temperature will favor, if the reaction is endothermic, if you decrease the temperature, will favor backward reaction. Hence, this is not the correct option. Next, higher concentration of N2. We know that N2 is a reactant. Increasing the concentration of the reactant will increase the rate of forward reaction, that is nothing but the formation of NO. Yes, this is correct. Next, higher concentration of O2. Yes, O2 is again one of the reactant. Increasing the concentration of reactant will increase the rate of formation of the product. So hence the correct one is D also. Let's see which option which has A, C and D. So that is option 2, A, C and D. So after finishing this question number 88, so we'll move on to question number 89. The question number 89 is from haloalkanes and haloarenes. Given below are 2 statements. Yes, statement 1 is benzene diazonium salt is prepared by the reaction of aniline with nitrous acid at 273 to 278 Kelvin. Yes, definitely. So if you take example, the benzene diazonium chloride. So this benzene diazonium chloride can be prepared by aniline between 273 to 278 Kelvin, that is 0 to 5 degrees Celsius, because if you use a higher temperature, it will decompose. Next, it decomposes easily in the dry state. Yes, so benzene diazonium salt, that is benzene diazonium chloride, will decompose in dry state. Except only one exception is benzene diazonium tetrafluoroborate, which is stable in dry state. So hence the correct statement is statement 1 is correct. Next, insertion of iodine into the benzene ring is difficult, and hence iodobenzene is prepared through reaction of benzene diazonium salt with Ki. Yes, definitely. So if you try to insert the iodine to benzene ring, we can do it by iodination. So iodination is reversible reaction. We don't get a good yield of iodobenzene. Hence we can use this reaction. So benzene diazonium chloride reacting with K, just shaking benzene diazonium chloride with potassium iodide, we will be able to obtain iodobenzene. So therefore both statement 1 and statement 2 are correct. So which is the option which has that? Both statement 1 and statement 2 are correct. Hence the correct option is option 3. Moving on to the last question. Very simple. That is organic chemistry question. It is from the chapter aldehydes, ketones and carboxylic acids. and also to some extent alcohols. Now let's see. So this is a compound which has a ketonic group and a nitrile group. Now this compound containing ketonic group and nitrile group is treated with Grignard reagent, that is methyl magnesium bromide. When it is treated with Grignard reagent, so that is excess of Grignard reagent, both the groups, that is CN as well as ketone group, can react with Grignard reagent. We know that whenever a ketone group reacts with the Grignard reagent, the ketone gets converted into a tertiary alcohol. Yes, so this CH3MgBr, if we react this with CH3MgBr, the two excess CH3MgBr, it gets converted into tertiary alcohol, that is OH, and then this CN will get converted into an intermediate which will undergo hydrolysis to give a ketone. So which is nothing but C, here you are going to get C, then CH3, then double bond N Mg Br. This on hydrolysis will undergo hydrolysis to give ketone. There also hydrolysis will take place, forming alcohol. So then you are going to get C double bond O, then CH3, then here OH. This on reacting with, this will again react with the ketonic group here, so resulting in the formation of a tertiary alcohol. Hence the correct option for this is, so here OH, then you are going to get here OH, CH3, and one more CH3 will be added. So the one which has the this option is option 1. So thank you, dear students. Now we have Mala madam from Department of Biology. She will take over.
Hello dear students, I am Mala, Department of Biology. Today we are going to solve last set of questions from biology, from question number 166 to 180. So here is the question. It's a match the following type of question. Match list 1 with list 2. List 1 is given, list 2 is given. We need to match this and find out which is the correct match out of this options. Coming to the first one, scutellum. Scutellum is nothing but cotyledon of monocot seed. You have already studied about it. Non-albuminous seed is example for groundnut. So it is B. And epiblast is rudimentary cotyledon. So which is C. Perisperm is persistent nucleus. So which is D. So which means that A is 2 and B is 3, C is 4 and D is 1. So the right answer for this question is option 3. We will go to the next question. Find the statement that is not correct with the regard to the structure of monocot stem. Here they asked not correct. So related to what we have to see, monocot stem, the options given are vascular bundles are conjoined and closed. Yes, it is related to monocot stem, which is a right statement. Phloem parenchyma is absent. This is also related to monocot stem, which is right. Hypodermis is parenchymatous. So this is wrong related to the monocot stem, because hypodermis in the case of monocot stem is sclerenchymatous. So the right answer is option 3, but still we'll counter check the other option which is there. Vascular bundles are scattered. Yes, this is related to the monocot stem. So the right answer which is not correct related to monocot stem is option 3. So we'll go to the next question. Twins are born to a family that lives next door to you. The twins are a boy and a girl. Which of the following must be true? We have to find the true statement here. The first option given here are they were conceived through in vitro fertilization, that is IVF. It need not be with the IVF, so this option is ruled out. They have 75% identical genetic content. Now it is not 75%, 50% chances are there. So this option is also ruled out. They are monozygotic twin. This is wrong. Why? Because it is one boy and one girl. It need not be a monozygotic twin. It's supposed to be dizygotic twin. So this option is ruled out. Last option which is given is they are fraternal twins. Yes. So fraternal twins means it is dizygotic twin. So which is nothing but two different sperm, two different ovum, two zygotes will be formed, one boy and one girl. So the right answer for this question is option 4. We'll go to the next question. Sweet potato and potato represent a certain type of evolution. Select the correct combination of terms to explain the evolution. Options are given here. So as soon as you see the sweet potato and potato, the function is same. The function is to store the food, storage of food, but the structure is different here. Why? Because potato is a root modification and potato is a stem modification. The structure is different here, the function is same, storage of food, which means that it is analogous organ, that is analogy. If it is analogy, you know that it is convergent evolution. So the right answer for this question is option 3. We'll go to the next question now. Which one of the following phytohormones, that is plant hormones, promotes nutrient mobilization which helps in delay of leaf senescence? Two characteristics is given here. Which hormone we have to find out? The options given are ethylene, abscisic acid, gibberellin, cytokinin. Coming to the word promotes, so it need not be inhibitor hormone. So wherever the inhibitor hormone is there, we will be ruling out that option, and this option is also not right. The right answer is cytokinin. It's a direct line from the NCERT. Cytokinin will help in promoting the nutrient mobilization and also help in delay of leaf senescence. We'll go to the next question. Why can't insulin to be given orally to a diabetic patients? So options given are human body will elect strong immune response. This is not the reason why they are not taking orally. It will be digested in gastrointestinal tract. Yes, because the insulin need to be digested with the help of digestive enzymes in stomach and intestine. So this is right. Let's cross check the other options also. Because of structural variation, no, this option is not related at all. Its bioavailability will be increased. This is also ruled out. The right answer for this question is option 2. It will be digested in gastrointestinal tract. We'll go to the next question. Name the class of enzyme that usually catalyze the following reaction. You have studied the concept called biomolecules, where you study the different classification of enzymes. So the options given are transferase, hydrolase, ligase and lyase. So here S stands for a substrate. They have already mentioned in the question. G stands for a group of other than hydrogen, that is also mentioned. S ash is also mentioned, that is another substrate. So if you see the reactant and the product, S, G, S ash is there. Here S, S ash, G is there, which means that S and S is combined and the G is shifted to the another place. It is transferred. So, as the name says that it is transferring, the enzyme which is involved here is transferase. So, the right answer for this question is transferase. We will go to the next question. It is a statement based question. Given below are two statements. Coming to the statement 1, the DNA fragments extracted from gel electrophoresis can be used in the construction of recombinant DNA. This is the statement 1 they have given. Yes, it is right, because DNA fragment is separated using the gel electrophoresis. The statement remains correct. We will go to the second statement. Smaller size DNA fragments are absorbed near anode wells in the agar, while the larger fragments are found in the wells in an agarose gel. This statement is also correct. You have studied that DNA is a negatively charged and they move from negative to the positive end. The smaller end will be absorbed in the anode, larger will be found near the agarose gel. So, both the statement are correct. So, let us see the option. Statement 1 is correct, statement 2 is incorrect. We will be ruling out this option. Statement 1 is incorrect, statement 2 is correct. This is also wrong. Statement 1 and 2 are correct. This is right. Statement 1 and 2 is incorrect, which is wrong. So, among these options, the right answer for this question is option 3, because both the statements are correct. We will go to the next question. The correct sequence of events in the life cycle of bryophytes is. So, here the question is related to bryophytes. The first statement given here is fusion of anthrozoid with egg, attachment of gametophyte to substratum, reduction division to produce haploid spores, formation of sporophyte, release of an anthrozoids into water. So, we have to find the correct answer from the options given below, and we have to arrange in a correct sequence. So, coming to this, so the first one is attachment of gametophyte to the substratum is the first step, and next is release of anthrozoids in water, and third one is fusion of anthrozoid with egg. The fourth one is formation of sporophyte, and the last one is reduction division to produce haploid spores. So, in this order, if you find, the right answer for this question is option 3, B, E, A, D and C. We will go to the next question. Genes R and Y follow independent assortment if capital R, capital R, capital Y produce round yellow seeds, small r, small y produce wrinkled green seeds. What will be the phenotypic ratio in the F2 generation? In the question only they have given a clue, that is independent assortment, which means that it should be dihybrid cross. And the question is, what is the phenotypic ratio in the F2 generation? It is very direct, you have studied that, 9:3:3:1. So, the right answer for this question is option 1. Next question. Each of the following characteristics represents a kingdom proposed by Whittaker. Arrange the following in increasing order of complexity of body organization. So, here if you see in an increasing order, it has to be from Monera to kingdom Animalia. Let us see from Monera to Animalia, and later we will match with correctly. So, coming to the first one, okay, prokaryote with cell wall made up of polysaccharides and amino acid is related to Monera. So, this comes first, and second one is eukaryote with cellular body, which is related to Protista. So, this comes second. Third one here is multicellular heterotrophs with cell wall made up of chitin, which is related to fungi. As soon as we see chitin, we remember fungi, right? Yes, it is related to fungi, which comes third. And fourth one, eukaryote autotrophs with tissue or organ level of body organization, which is related to plantae, which stands fourth. And the last one is heterotrophs with tissue or organ system level of organization, that is related to Animalia, which is fifth statement. So as per this order, the right answer for this question is option 4. We'll go to the next question now. It is a match the following type of question. Match list 1 with list 2. Here the list 1 given is centromere, cilium, cristae and cell membrane. You have studied in the cell chapter. Coming to the centromere, they help in cell division. Cilium, it is there for the movement, cell movement. And cristae is a part of mitochondria, so which is C. Cell membrane has phospholipid bilayer. So in this order, if you find, the answer is A is 2, B is 3, C is 1, D is 4. So the right answer is 2, 3, 1, 4. Fourth one is the right answer. We'll go to the next question. Which one of the following equation represents the Wellhurst-Pearl logistic growth of population? It is a very direct question. They've asked you the equation of this Pearl-Hurst logistic growth of population. The right answer is dN by dt is equal to Rn k minus n by k. The right answer for this question is option D. We'll go to the next question. It is a match the following type of question. Match list 1 with list 2. Here emphysema they have given. Emphysema you have studied related to like respiratory surface. A is 2. And angina pectoris you have studied about acute chest pain. Glomerulus filtration, it is related to the kidney. Tetanus related to the muscle. So A is 2, B is 3, C is 4, D is 1. 2, 3, 4, 1. The right answer for this question is option 4. We'll go to the last question in biology. Cardiac activities of the heart are regulated by nodal tissue. Yes, it is controlled. So nodal tissue is right. A special neural center in the medulla oblongata. This is also right. Adrenal medullary hormones. This is also right. Adrenal cortical hormones. It is medullary hormones means cortical is not right. So A, B and C are right here. So the right answer for this question is A, B and C only. Third option is right. So the further question will be discussed by physics teacher. Thank you.
Yes, welcome to the last segment of the physics. Yes, my name is Sudhir and I'm done BTech from IIT Kanpur. I'm a senior lecturer and having the experience of 5 years. Now if you see the question number 31, in this question it is based upon the errors, especially percentage errors. And if you see here they have given in the terms of what? Powers and multiplication and division. If that is the case, how would we do? We'll write percentage error in P, means percentage error in the LHS is equal to what? We take the power into percentage error of each quantities that is present on RHS. So it is percentage error in P is what? What is the power there for A? 3 into percentage error in A plus 2 into percentage error in B, and what is the power for C? Power 1 plus 1 percentage error in 1 into percentage error in C plus what is that power for D? It is root D, means 1 by 2. So 1 by 2 into percentage error in D. So what is there? What is the percentage error in A? 1. So 3 into 1 plus 2 into percentage error in B is 3 plus 1 into percentage error in C is 2 and 1 by 2 into 4. I think if you calculate 3 plus 6, 9 plus and 9 plus 2, 11, 11 plus 2, 13. So 13% is what the answer here. So 13% is the answer here. Now next question, question number 32. If you see the question number 32, they have given the sun is rotating about its center and what is the time period? It is 27 days, but they suddenly told in the question the sun were to expand to twice its present radius, so without any external influence. So there is no external influence, it was bulged, means it was, the radius has become twice. Now they are asking you, sun is assumed to be, yeah, uniform density. Now how to approach this question is, there is no external influence, so I can take what, there is no any kind of external torque, so I can conserve the angular momentum. So in that process I will write here, let us say the initial case, it is I omega is equal to what? Constant. So initial moment of inertia is I1, and here what happens is, here they did not give you in the terms of what? Omega. They did not mention anything about omega, but they have given time period. What is the relation between omega and time period? 2 pi by capital T. So 2 pi by capital T1, and here I omega is constant. So what I am doing is I1 omega 1 is equal to I2 omega 2. I2, this is the moment of inertia when it is expanded, and what is the time period they are asking after the expansion. So T2. So 2 pi will it cancel out. I think so we will get like this, T2 is equal to, so we will get T2 is equal to, so I2 by I1 into T1. So this is what we get, I2 by I1 into T1. But you know it is a, what I can say is, it is, if you see clearly, I2 by I1 into T1. So the sphere is what? Solid sphere. The sun is what? Solid sphere. So I can say that, I think so it was expanded. So, and I can clearly say it is what? Moment of inertia. Moment of inertia, it was expanded. Fine. So with that twice of its radius. So what will happen to the radius now? So I can say I is proportional to what? R square. So you will get R2 by R1 square into what? T1. So what is R2? Twice of it. So R2 is twice of it. So if you substitute the value, R2 is equal to 2 R1, so you will get 4 into T1. So what is the T1 value? 27. So T1 value is 27. What is the 4 into 27? It is 108. So that is the answer. Answer is what? 108. Now question number 33. If you see the question number 33, they are talking about radius of a orbit, means they are talking about circular orbit. They are talking about what? Circular orbit. Means we know the relation between the time period and circular orbit, right? So T is what? Proportional to radius power 3 by 2, right? So here what I am doing is, they have given you two planets, one is Martian, another one is Mercury. So I will put the Martian as a, I will give the number 1 and Mercury for the number 2. So what I do, they are asking about what is the length of, means they are asking about what is the time period of the Mercury. So I will write T2 by T1 is equal to R2 by R1 whole power 3 by 2. Got it? Now what I have to find T2. So here T2 is equal to what? R2 by R1 whole power 3 by 2 into T1. So here what is the T1 here? T1 is Martinian is the one, right? So that is the T1 is 687. And what about the R2 and R1? Here you see they have given the relation like this ki R1 Martinian orbit is what? 4 times the what Mercury orbit. Means they have given R1 as a 4 times of R2. So ultimately what I get here 1 by 4 I will get. So 3 by 2. So if I solve this, what will I get? So I think if you solve 4 power 3 by 2 like that, you will get 687 by, you will get something like 8. So approximately if you calculate, you will get 88 Earth days. So option 3 is the answer for this. So the next question here, a wire of resistance R. See, remember thing we had taken one wire, right? R is equal to what? Rho L by A. So if you are taking some wire, obviously the rho value, because it is the same wire, rho value, and it is the same cross section, so A value will be the same. Even if you cut it, the rho and A will be the same for each part. So simply I can say R is equal to rho L by A and R is proportional to L. So if you are cutting 8 parts, means each part length becomes now what? L by 8. So R will become what? R by 8. So each part R is what? R by 8. Now what you are doing is you are taking 4 of them, 4 of the parts, and you are connecting in parallel like this. And next another 4 parts you are joining as a series. Now here you see, so now what is the effective combination they are asking it. So what is this? R by 8, all are R by 8, let me say R dash, all are R dash, R dash. So R dash everything, that is what? R by 8. Now if you see clearly parallel, the same equal resistance parallel are there, what is the effective is? R by N. If the N identical resistors are in parallel, R by N. So this one will become what? R dash by 4, and another one part will become what? R dash by 4. And what is this? This both are in series. So you just add them. So it will become ultimate equivalent what? R dash by 2. So what is the R dash? R by 8. So what is the answer? R by 8 by 2, that is R by 16. So this is what the answer here. So I think it is doable question. Next question, if you see here the photon is a massless, electron is a having the mass. Okay. Now they are asking about the what? De Broglie wavelength ratios. So how would you write? Let us say for example in the proton the wavelength is written as what? H by E by C, because the momentum is what? E by C, and for electron it is H by, if I want to write in the terms of energy, the momentum can be written as root of 2m E, root of 2m E. So HH will get cancelled out. So if you do, I think so, the C will go up, C it will becomes like a into C, and it will becomes root ME by E. So ultimately what will be the option? If you take root E and root E times, you will get the option 1. Next question, 36. Now you see this question is little bit of related to the moment of inertia, but we have to take care about how the masses to be taken in this question. If I want to say here, what they are asking is, they have taken a larger sphere, solid sphere, and its radius is what? 2r. Okay. Let us say what is the moment of inertia of the larger sphere is, let us say it is I1. It is a solid sphere, right? So it is 2 by 5 mass into r square. So 2 by 5, I think let me take the mass as a m, but radius square is what here? 2r square.
So ultimately what you are getting it here, if you calculate, it is a 4r square, it is 8 mr square by 5. This is the I1, this is for the whole solid sphere. So now I am taking I2 as the small portion, means which is removed, this portion. But the problem here it is, here see, moment of inertia is taken exactly the central axis. That is why we have used the formula 2 by m mass into radius square. But if this small piece, if you see this axis is what? Tangential axis. So I think so I am directly taking the formula. You can use the the parallaxis theorem and you will get something like 7 by 5 mass into radius square. But the mass here very important to be noted here. Here the mass, the total mass is capital M, but this portion mass is actually something part of that capital M. So I will write like this key, what? 7 by 5, that portion mass is m dash, and what is the radius for this? R square. But what is that portion mass? How do you know that that portion mass is? They have told it is a large solid sphere. So if let us say they are not mentioning any kind of thing, by default you have to confirm it is a uniform mass distribution. So if let us say if I do the here work, how to find this m dash, you just what you have to do is you have to take the density and you have to, it is a uniform density case. So total mass by, what is the total radius? Something like 4 by, what is the total volume of the sphere? That is 4 by 3 pi. The radius is 2r is the total radius of the, this is for entire sphere. For the portion m dash 4 by 3 pi r cube. So if you do the calculation, you will get the result as a m dash as what? M by capital M by 8. So that is what it becomes, 7 by 5 capital M by 8, and this is what? R square. This is what I2. This is what the moment of inertia of the removed portion. But here the moment of inertia is what? It is a tangential axis. That is what you have to take in care. Now next one is they are asking about rest. See, usually what will happen is the moment of inertia, you can write like the moment of inertia of the whole body is equal to what? The sum of the moment of inertia of its parts. So that is what I am doing it. If let us say they are asking about this part, so what you do? If I want to do for this part, so what you do is you will take the moment of inertia of the whole sphere I1 and this removed sphere I2. So you will get I1 minus I2. So this part, especially this one, this part I want to know. So now what you get now? I1 minus I2, that is the rest part. And they are asking about ratio of the smaller as well as the rest part. So what does it mean? They are asking about the ratio here. They are asking about the ratio I2 by what? That is the smaller part is I2, I1 minus I2. So you have substitute the values now. What is I2? 7 by 5 capital M by 8 R square by, what is the I1 value? 8 mr square by 5 minus 7 by 5 m by 8 r square. So this is what you will get. Fine. So now if you cancel out mr square, which is the common, and if you do the something like, you will get 7 by 5 here divided by, I think so here 8 is there, so be careful, 8 by 5 minus something 7 by 40. If you do the LCM and if you take if you do the LCM, so you will get 7 by 57 as the answer. This is the answer. If you do the LCM, you will get this answer. 7 by 57 as the answer, you will get it. So this is the 36th question, little bit, yeah, lengthy is there. Now 37th question, if you see properly 37th question, see if you see the 37th question clearly. So what will happen here, it is they have given electron and they are saying that it is injected into what? Magnetic field perpendicular, and they are saying that electric field is also what? Present there, uniform, all are uniform, uniform magnetic field and uniform electric field. So but they are saying that ultimately what is happening is it does not getting deflected. See what is the case it happens, let us say, if let us say this is the magnetic field, let me take the magnetic field horizontally, this is the B. magnetic field horizontally. And if let us say for example, if you let us say you will just horizontally. So, what will happen if let us say for example, if the something like electron is entering here, with the V, with the electron is entering V like this. So, you will get the force, right? How would you get? You take the V cross B. So, it is into, that is for positive charge. So, for the electron what will happen is, see what is the force on a charge? It is the force on a charge direction is decided by V cross B, that is into, but electron is what, negative charge. So, you have to take the negative direction. So, what will you get? The force is something like outwards you will get, something outwards you will get. This is what force you will get, right? But it is not getting deflected, means what? There should be some kind of opposite force should present, means there should be some force opposite to magnetic force. It should be what? Something like electric field, right? Electric field. So, how the electric field should act? Obviously, electric field should act outwards. If the electric field act outwards, the electron will move into, means the force on the electron will be into. So, that magnetic force out, Fb out and Fe into, both will balance. So, what is happening? If I take the B horizontal, electric field should be what? Outside the screen. Okay, outside the screen. So, electric field should be what? Outside the screen. So, obviously they are perpendicular. For the things to happen, the electric field should be perpendicular to B. But here, which option is correct? Which option is correct here? Why? Because 3 and 4 both are telling, but magnitude how do you find? Because the forces are equal. So, I can write what is the force? QVB. That is the magnetic force when the charge enters perpendicular, and it is equal to what? Electric force. So, both are equal. Now, you substitute the values. I think so B value is given and V also given, the speed of the electron is given like a C by 100. So, you substitute C by 100 and into what? What is the B value here? 9 into 10 to the power of minus 4. So, that is what the E value. If you do the calculation, put the something like 3 into 10 to the power of 8, you will get 27 into 10 to the power of ultimately 10 to the power of 8. If you do the calculation, I think so you will get something around 27 into 10 to the power of 2 volt per meter, which is the SI unit of the electric field. So, option is what? 4 here. Option is what here? Option is 4 here. Now, I think so it is the formula oriented, but you must clearly, if you draw the diagram, either things will become sorted out. Now, you take the question number 38. In the question number 38, observe the equation first of all. Ez is given, means the electric field is along the z axis, along the z axis, z axis, whatever it is. Now, coming to the point here, it is x is given. So, it is talking about wave propagation, means EM wave propagation, that is x. But what is positive x or negative x? You check the coefficients. If the coefficients are of the same sign, it is 5 and this one positive, same sign means it is a negative x, or I can negative x or minus I, that is what the wave propagation direction is, minus I cap. Now, what about the so called B? How would you find the direction of the B? You know that the E cross B is equal to what? C cap. What is the E direction? Z, that is a K cap. Next one is what? What is the B direction? That is a fill in the blank like that way. So, minus I cap. So, if I fill which direction, I will get the minus I cap, J cap. So, I can say that B is what? Belongs to what? Y. But here if you see, okay, you should get the same cos, means same cos is there, but how would you find the amplitude? Whether, I think so, you actually you can find the amplitude also by using what? The E naught is equal to C B naught. So, what is the B naught? E naught by C. What is the E naught? 60. So, what is the C value? 3 into 10 to the power of 8. So, if you, I think you will get the answer something like 2 into 10 to the power of minus 7. Yeah, and it is a Y. So, answer is what? Option is what? Option 3 is correct. Option 3 is what? Correct. Yes. Now you see this question, I think so. This question, question number 39. It is the fundamental question. It is from NCERT. If you see clearly, see it is a NCERT based question, and there is a direct question with the numerical changes in the NCERT, even back exercise. Weight is what? 48 on the surface. But what they are asking, gravitational force. What is a gravitational force? It is weight itself. So gravitational force experienced by the body due to earth at a height equal to, see here, this is very, very important. It is a case of what? GH, the gravity at height h case. But the problem is which formula you have to use. If you see the height h is 1/3 of the radius, means it is not much lesser than the radius. So you have to use this formula, g by, this is the g on the surface, 1 plus h by r square. Now what they are asking, weight. Weight means just to multiply with m on both sides. So this is the weight at height h, this is the weight on the surface by this one. Now what is the h value? R by 3. What is the h value? R by 3. Substitute. What will this becomes? Mg by 1 plus h by r, that is r by 3 means 1 by 3 you will get it. And what is the mg value here? Mg value is given as a 48. So 48 by, and this one if you do it is 4 by 3 square, I think you will get something like 16 by 9. So if you calculate 3 times and 3 9 times is what? 27. So option 4 is the answer. Option 4 is the what? Option 4 is the answer here. So I think it's right from NCERT. Now you see this question. An unpolarized light beam traveling in air, they have told it is what? Brewster angle. What is the Brewster angle? Means if you hit with the Brewster angle, what will happen is, this is the surface, this is the interface of two medium, and it will hit with the Brewster angle. The reflected will be completely polarized and refracted will be partially polarized. So what is the relation Brewster angle? Let me theta b, that is equal to what? Tan inverse of mu. Mu is what? Refractive index. What is they have given? 1.73. Here you 1.73, actually the people will get stuck here. 1.73 is matches with the root 3. So what is the tan inverse of root 3? 60 degrees. That's why 60 degrees. I think I can say closer actually. So close to 60 degrees I can say, because that 1.73 also closer to what? Root 3. But you know theta b is what? Brewster angle, which is angle of incidence. But you know the angle of incidence and angle of reflection is what? Same. So this will also be theta b. So this will also be closer to theta. So if you check the option, anything is there? I think so. The option 3, the reflected light is completely polarized, and angle of the reflection is what? Close to 60 degree. So I think so. It will be, yeah. So option 3 is the correct one. Yes. Now you see question number 41. Here the main thing is they are asking about, they have given the circuit, it is straightforward, no need to describe again. Okay. So they have given this 1 ohm, 2 ohm like this way. Now how to find the current here? CD. Fine. I am taking like this is the total current I, and it is distributing I1, and this one is I2, and this one is, let's say for example I1 is, it is there, and this one is I3. I3 is what? Current through the CD. So what I need to find? I3 value. So this one is what? If the I1 is coming out from the across the one through the one, and it is distributing as a I3, and next one is what? I1 minus I3. And what about this? I2 is coming from the 3, and I3 also coming through the CD. So it will be I2 plus I3. What I require to find? Now what I require to find is I3. How do I find? See, listen very carefully here. You should understand how the circuit things work out. Here you see the C and D points, right? If you see clearly, the current is passing through the C and D. I am not saying it is not passing through, but if you see that it is a conducting wire. So conducting wire, there is no resistance, so I can say the voltage is same here. Okay, I can make it like a single point. I can make it like a single point. Like that I can make the circuit. Now what will happen here? This one is 1 ohm, this one is 3 ohms, this one is 2 ohms, this one is 4 ohms. Fine. Now what will happen? I will find the equivalent now, equivalent resistance. Okay. From the equivalent resistance, I will find the current, actually I current. What is the current through the circuit? So now 1 and 3 is what? Parallel. So 1 and 3 parallel, I can just use that R1 R2 by R1 plus R2. This one is also parallel, ultimately both are series. So 1 into 3 by 1 plus 3. What is the value? What is the value? 3 by 4. And here it is what? 2 into 4 by 2 plus 4. What is the value? I think so you will get something like 8 by 6. I can write 4 by 3. Both are in series. So what is the equivalent? 3 by 4 plus 4 by 3. So if you solve it, I think you will get something like 20 by 5, 25 by 12. 25 by 12. You will get 25 by 12. That is what equivalent. 50 is there. So what is the current? V is equal to IR. If I find V is equal to IR, I will get the current value. So I will get the current value. So I think I will add, so slide here. So V is equal to IR. So I is what? V by R, that is 50 by 25 by 12. So what I have got, I value is what? 24. I got I value is what? 24. Now how do you do this with I value? Fine. Now let's see. I have got I value is what? 24 here. Now if I got I value 24, now if you clearly see in a method, that one is, this one is 1 ohm, this one is 3 ohms, but if you see clearly that this voltage across the 1 and voltage across 3, what? Same, it is same, because the same point will be there, A here, here also the same point, it will be there, voltage will be the same. So what I do now here, it is, so I can write the voltage is same, the current, how the current will distribute, how the current will distribute. So the current will distribute is, here what we have got, I think so what we have got here, I value is 24. So how does it distribute? So here the voltage is same, 1 ohm, next one is what? 3 ohms. So how do we get that? How do we get that? I am just redrawing the diagram because the space is not enough. So 4, like that way we are getting, right. So how do I get? See, it is like a 1 and 3 is there, right. So this is the 1, this is the 3, but you know the current is inversely proportional to resistance. So what will happen here? The 3 parts of 24 will go to 1 ohm, because it is a 1 by 3, right. Here this is the resistance is what? 1 part. The resistance wise it is a 1, it is a 3. So current wise it should be reverse. So 3 times of the current should go for 1 ohm and 1 part of the current should go for 3 ohms. So I can say clearly here that one is the I1 and I2 is there. I1 is what? I1 value is what? 18 ampere, and here it is what? 6 ampere. Fine, 18 ampere and 6 ampere. Now how could I get the I3 now? This is the I3. How could I get it? I3. So here you see, this one is what is the current flowing through, this one is I1 minus I3, and this one is what? I2 plus I3. Here also I can say that 2 ohms and 4 ohms are what? Parallel. So if I just, what I do here, it is, so I just parallel, so I am just equating the voltages. I will get 2 into I1 minus I3 is equal to 4 into I2 plus I3. So if you substitute the values I1 and I2, you know, right. Now 18 and 6, if you substitute the values, you will get I3 value as what? Substitute the values, because I1 and I2 I know. So I3 value I will get what? Answer as a 2 ohms. means without the KVL we can do like this also. Yes, answer is what? 2 amperes. This is the what we have done the work. Next, fine. Now let us go to the next question. This one is 42nd question. See here, it is a little bit lengthy question. What is happening here? It is initially you have taken one capacitor. Initially you have taken one capacitor, fine, without any filling. Obviously without any kind of dielectric, what is the capacitance value? Epsilon naught A by D. Now what you have done is, initially is what? Epsilon naught A by D. This is what they have told that. They have told that the plates are separated by D, and now after what happened is, now you are taking 2 slabs of different dielectric constants, and now you are filling them. Now you are filling in the space between the plates of a capacitor. So now what will happen? The capacitance will become twice. Now this is the where we have to concentrate more here. It is this is now you are filling with the space, something like, they did not tell how they have filled it. Maybe there may be a gap or may not be. So 3D by 8, D by 2 and 3D by 8. Sorry. Now this is what we are getting it, right? Now one is what? 3D by 8. Another one is what? D by 2. Now due to this the capacitance becomes twice. Now what is this equivalent capacitance is? It is a twice of this. But if you see clearly here, we can take like this 3 capacitance parts. One is with the dielectric 3D by 8, another one is D by 2, another one is D by 2. So another one, whatever it is there, next one is vacuum, that is you can take like a third capacitance, but all are in what? Series, all are in like a series. So what is the next space that is remaining? See, D by 2 and 3D by 8 is there. Total space is what? Total spacing, what is the separation between the plates is D. So if you do something like D minus this one 3D by 8 plus D by 2, so overall what you get here, it is you will get 3D by 8 and D by 2, you will get what? D by 8. So this is the, what I can say, it is filled with no dielectric. Fine. So I am taking like a 3 dielectrics. One is with the 3D by 8, D by 2, another one is no dielectric is D by 8. So all are in like a what? Series. But what I think, so 1 by C effective or C equivalent is equal to what? 1 by C1 plus 1 by C2 plus 1 by C3. Next, 1 by C effective is what? I think so they have told that it will be twice of the earlier case. So twice of means 2 epsilon naught A by D will be there. So you will get D by 2 epsilon naught A. What about the 1 by C1? Now you see very carefully, the C1 is what? I have taken the K1 constant, the dielectric K1 capacitance is C1, K2 is what? C2. But what is that thickness for K1 is 3D by 8, thickness for this, what is this? D by 2. So what I use the formula is K epsilon naught A by D, but here D is what? Something else, 3D by 8 and D by 2, like that way. So I will get ultimately, if you see D by, ultimately what I get is D by, this one is D by, I think so 3D by 8, that is what the D here, right? That is what the thickness, 3D by 8. So K1 epsilon naught A. And what is the thickness here for the K2 is D by 2, that is K2 epsilon naught A. What about the C3? C3 is just like a what? Without dielectric. What is the thickness there? D by 8 by epsilon naught A. Now cancel out all epsilon naught A and D also you cancel out. fine. So now ultimately if you solve, if you want you can take that K2 value as a 1.2 by K1, 1.2 by K1. So what will happen here, this one is 1 by 2 is equal to, next one is 3 by 8 K1 and 1 by 2 K2 is there, but K2 I am writing like a what, K1 by 1.25. So 1.25 will go that side, it will become 1.25 by 2 K1 plus what, 1 by 8. Just to solve it, I think so you will send the 1 by 2 minus 1 by 8, and if you solve it, so I think so the for easy I can write like this, this is the 5 times and this is the 8 times. Yeah, fine. So you will get what, 8 by 8 K1 plus 1 by 8. If you solve it, you will get something like K1 as a what, if you solve it, K1 as a, you will get something like 8 by 3. So the answer will be what, 2.66. So this is how we have to solve approach. The next question, if you see this question, they have described lot of things ki 10 vernier scale division will coincide with 9 main scale divisions, and they have told that the least division in the main scale is what, 0.1 centimeter. Now here the main thing for doing is they have given the MCR also, main scale reading is 5 and coinciding vernier scale is 8. So we need the least count. How do you find the least count here? Least count is 1 MHD main scale division minus 1 VHD. Okay, but you know 10 vernier scale divisions is equal to what, 9 MHD. So I can also write like this ki 1 VHD is equal to what, 9 by 10 MHD. So if I substitute 1 MHD minus 9 by 10 MHD, so what would I get here, 1 by 10 MHD. So what is MHD? Main scale division. 1 main scale division, what is they told, 0.1 centimeter. So 1 by 10 means I will get 0.01 centimeter as a what, least count. That is what the least count here. Now if you see how do you find the reading, MSR plus VSR, MSR plus VSR, and correction should be done. Okay, how the correction should be done I will tell you, because they have told that key correction is required because 0 of VS is, so means what is the meaning is, when the jaws are not closed, when the jaws are closed, it's fine, there is some equipment is present there, you are measuring something, but jaws are closed means still it is showing some reading, means it is error. So it is showing some error 0.1 centimeter, means extra reading it is showing. So you have to, whatever you do the MSR plus VSR, ultimately you have to remove that. So because extra reading it is showing, right, actual should be what, you have to remove that, whatever the error it is showing. So minus 0.1, that is what you get. So what is MSR? 5. Okay, fine. What is the VSR here? VSR is what, the number of coinciding division, which is 8, and 8 into 0.01 minus 0.01, 0.01 minus 0.1, and you will get ultimately what, 4.98 centimeter. If you calculate, you will get this answer, this as the answer. Fine. Now let us see this question. See, in a circular coils, what is the magnetic moment is number of turns into current into area. Here only one coil they have taken. So different small circular copper, means they have taken the 2 coils, but number of turns is what, they did not mention. So means it is only 1 it is representing. Okay, fine. So what I can write is M1, they are asking about ratio of the respective magnetic moments. Now N1 is equal to N2 is equal to 1, that is fine. So I am just writing I1 A1 by I2 A2, but they told that in the information the same 2 ampere current is flowing through the coil. So what does it mean? So the current is same, so I can cancel out. Now A1 and A2 is the coil, it is a circle, obviously what is the area? Pi r square, pi into radius square. So I can just write radius square ratio. So radius square ratio is what? R1 by R2 square. So what is the, I think radius ratio they have given 1 by 2. So 1 by 2 square is what? 1 by 4. 1:4 is the answer. Fine. I think directly formula oriented question it is. Yeah, now this is the question, hot topic question. See, one important thing that I want to say here it is, here there is a water tank, some water is there, and here there is a some wall. In the question they have described there is a one wall is present at x is equal to L, and the wall is like something like it is intersecting this board, okay, z direction it is there, when filled with a liquid of some surface tension and some density rho. So x is equal to L. So that is what here it is. Now how do you solve this question is, see if you see clearly, this one is like a cylindrical surface kind of. If you see clearly the surface of this water surface, it is a cylindrical surface. There is one direct result is there that will help you. See, if let us say this is the, if you just go point here, this is a P naught, means because it is exposed to what? This is a P naught, the atmosphere pressure here it is what? Just above this or just the surface is P naught. Just below it, if any cylindrical surface is present, so there what will happen is, if you see this is the concave part, right, concave part will be having the excess pressure compared to the convex part. So if this is the P naught, and it will be how much, what is the excess pressure value is surface tension by what? Surface tension by radius of curvature. So the P naught will be having the excess, how much it is excess? S by R. So obviously if it is a P naught, if it is a P naught, so obviously below that point should be what? P naught minus S by R, radius of curvature. Yes. Now coming to the point, what we require it is, and if you clearly see this point, you just observe this, this is exactly P naught. This is a P naught, right, exactly just touching the, almost like it is on the atmosphere. If you relate this both points, let us say this point and this point, so obviously if you see that, if I use that pressure variation along the vertical, I can say this P naught minus, let us say this is the P1, so P1 is equal to what? P naught minus rho gy, right, P naught minus rho gy, because as you move along it will reduces with rho gy, right. So that what I can take. Now I have told you for cylindrical surface there is a result, the concave portion will be excess pressure with the amount what? S by R. So obviously convex will be what? Minus S by R. I will equate them. Now what happened here, if you cancel out P naught and P naught together, so you will get one result, that is, I think rho gy is equal to S by R. Let me write here. Now what is the R? Now R is radius of curvature. See here, if you clearly see in the options, they have given in the terms of what? Second derivative, d2y by dx square. So there is one relation with the radius of curvature and this d2 and this second derivatives and first derivatives dy by dx and d2y by dx square, that is R is equal to 1 plus dy by dx square whole power 3 by 2 by d2y by dx square. Fine. Now what here told in the mention, in the question they have mentioned that theta is what? Dy by dx, okay, but it is very less than 1. So what I can do is, this one I can ignore, I can neglect compared to the 1. So now what the things will become, the R will become what? 1 by d2y by dx square. Fine. Now if you substitute here, so I can say clearly ki R is equal to 1 by d2y by dx square, means d2y by dx square is 1 by R. So now this term will become what? Rho gy is equal to what? S into d2y by dx square. Now what is the answer now? If among the options, if you send the s to the other side, d2y by dx square will be what? Rho g y by s. Yeah, little bit tricky and also a bit high level question, I can say, matching with the JE. Now what I want to say here, it is clearly, see, I understand if you see the questions of the physics, it is a tougher level, but what I say is ultimately the competition, you are facing the NEET, it is a competition, right? All the things are in the relative manner. Means if you take the word of the students in this exam, especially NEET 2025, it is tough, relatively to all it is a tough. So you think like that way, positively, relatively it is a tough. So now the exam is done, little bit you relax yourself, I think so. I am also just, you have to go well, and all the best for the future endeavors. And one more thing, I want to go one thing, that is, see, RV institutions is launching the course NEET Replay, that is for full time NEET repeaters program, and it is aiming for the NEET 2026 and based in Bangalore, and the classes starts from June 2025. If you want to contact, or if you want to know any details about that program, you can contact to this number, and the location is based on Alanka and Bangalore. I think like, comment, share and subscribe the RV Learning Hub channel. Yes, thank you all, and all the best for your future endeavors.